Imagine you've just been handed this vial of cerebrospinal fluid.
High-stakes stuff right there.
Exactly. I mean, you have a patient lying in the ICU, their life is entirely on the line, and the physician is just waiting on you.
The pressure is definitely on.
Right. So you set the vial on your bench, and you realize your media plates aren't quite ready yet.
Oh, that's a classic lab scenario.
It is, right? And your immediate instinct, the same instinct you use for like milk or leftovers, might be to just pop that sample in the refrigerator to keep it fresh.
Which would be a massive mistake.
A completely catastrophic mistake.
Yeah.
Because if you do that, you will have just murdered the very pathogen you're trying to identify.
Yeah, you'd get a false negative. The patient might get the wrong treatment, and it's entirely because of, a simple temperature error.
Which is wild. So welcome to this deep dive.
Glad to be here. That fridge scenario, by the way, is the exact reality of the clinical microbiology lab.
It really is.
Yeah, I mean, we are dealing with living, breathing, incredibly finicky organisms. A single degree of temperature or a slight shift in atmospheric pressure can literally make the difference between a definitive diagnosis and a fatal miss.
Right. So listen to me very carefully. We are locking the doors, turning off the notifications, and just clearing the desk.
Zero distractions today.
Exactly. You are here for an intense, zero-fluff ASCP MLS board exam audio cram session.
Pure high-yield facts.
Yep. Our mission today is absolute clinical ID mastery. We're talking media, stains, biochemicals, and diseases.
We are going to take your provided review document and just wring every single drop of testable information out of it.
And we aren't just going to memorize lists, right? We're going to really understand the biological mechanisms behind them.
Because the board examiners, they aren't just testing your memory, they're testing your clinical reasoning.
Right, they want to know if you understand why a specific test works.
Exactly, because that understanding is what actually prevents you from making those catastrophic errors at the bench.
So here is how we're going to attack this. We are covering 10 foundational and organism-specific sections.
And after every single section, we are putting you, the listener, right in the hot seat.
Yep, we're running a rapid-fire clinical case study styled exactly like the multiple-choice questions you will face on the exam.
We'll break down the deductive reasoning required to find the right answer.
And we'll explain the biochemical traps hiding in the wrong answers, too.
Which is crucial. And finally, we'll wrap up the entire deep dive with a lightning-fast cram pass of every major test alert box from the material.
Right. That structure is going to build the exact neural pathways you need.
So when you sit down for the exam, you shouldn't be guessing. You should be recognizing patterns.
Let's get into it. We have to start at the very foundation of microbiology, which is how organisms grow and why the lab manipulates these exact conditions to coax them out of a clinical sample.
Because you can't identify a pathogen or run a biochemical panel or test an antibiotic if you can't get the bug to grow in the first place.
Exactly. So, section 1, growth. Let's talk about the core requirements, starting with temperature.
Right. So bacteria are categorized strictly by their preferred thermal environments. You have your psychrophiles, which are the cold-loving bacteria.
And their optimum temperature is what, right around 15 degrees Celsius?
Yeah, exactly, 15 degrees.
Yeah.
Then on the opposite end of the spectrum, you have the thermophiles.
The heat-loving ones.
Right. And they thrive anywhere between 50 and 60 degrees Celsius.
But the sweet spot for the clinical lab, like the organisms we care about the most, are the mesophiles.
Yep, the mesophiles. Their optimum temperature is exactly 37 degrees Celsius.
Which, not coincidentally, is human body temperature.
The evolutionary adaptation there is just brilliant. I mean, the vast majority of pathogenic organisms fall into this mesophile category.
Because they've evolved over millennia to view the human body as a resort.
Precisely. They have tailored their entire enzymatic machinery to operate at peak efficiency right at 37 degrees.
So if you drop the temperature, their enzymes slow down.
Right, and if you raise it, their proteins literally begin to denature.
Which perfectly explains why our clinical incubators are strictly locked at 37 degrees.
Exactly.
Now, what about the pH of the environment? I know human blood is tightly regulated, right around 7.35 to 7.45.
It is.
Do the pathogens expect that same regulation on a plate?
They absolutely do. And this pH preference is actually a fantastic early differentiator between bacteria and fungi.
Oh, really? How so?
Well, bacteria prefer a relatively neutral pH, right around 6.5 to 7.5, But fungi, on the other hand, require a slightly more acidic environment,
Look what range.
hovering right around a pH of 5.0 to 6.0.
Oh, wow. So if a lab is trying to grow a stubborn fungus, they'll actually acidify the media.
Yeah, to give the fungus a competitive advantage while simultaneously suppressing all the bacterial normal flora.
That is the exact strategy. Now, to maintain these incredibly specific optimal environments in our artificial culture media, the manufacturers use phosphate salts, right?
Yes, as chemical buffers. Because as the organisms grow, they produce metabolic waste that could wildly swing the pH of the agar.
So the phosphate salts absorb those excess hydrogen or hydroxyl ions.
Exactly, keeping the pH stable so the organisms don't just poison themselves in their own waste.
Makes sense. Speaking of environments, let's talk about osmotic pressure. Most bacteria want an isotonic environment, meaning the salt inside the cell matches the outside.
Right, but the board absolutely loves to test the exceptions here.
Of course they do.
The critical exception here are the halophiles. Halo meaning salt, halophile meaning loving.
So these are organisms that actively require a higher salt concentration.
Yeah, to stabilize their cell membranes. The classic board-certified example is the Vibrio species.
Ah, Vibrio. So if a patient comes in with severe diarrhea after eating raw oysters and you suspect Vibrio, you must consider its halophilic nature.
Absolutely. Standard media might actually cause the Vibrio cells to undergo osmotic lysis.
They would literally absorb water until they burst.
Yeah, so you have to feed them salt.
Let's move to oxygen. I like to think of oxygen in a bacterial culture, like a highly toxic corporate work environment.
That's a great analogy.
Right. Oxygen isn't just air. It is a highly reactive molecule that wants to tear electrons away from other structures.
And the text specifically highlights the formation of toxic superoxide radicals when oxygen is metabolized.
It's essentially metabolic pollution that just shreds bacterial DNA in cell membranes.
That toxic workplace analogy is surprisingly accurate, yeah. Oxygen metabolism inevitably creates these highly reactive, damaging byproducts.
Because an unpaired electron in a superoxide radical is incredibly destructive.
Exactly. So if a bacterium is going to survive in that oxygen-rich environment, it needs a highly competent internal cleanup crew.
Like an HR department.
Right, to handle the toxic fallout before the cell dies.
And that biological HR department consists of two very specific enzymes, doesn't it? Catalase and superoxide dismutase.
Those two enzymes are literally the difference between life and death in an aerobic environment.
So aerobes, which absolutely require oxygen to survive, and facultative anaerobes, which are flexible, both possess these enzymes.
Yep. Superoxide dismutase acts as the first responder. It neutralizes those incredibly toxic superoxide radicals into hydrogen peroxide.
But hydrogen peroxide is still toxic.
It is. So catalase steps in as the second responder. It breaks the hydrogen peroxide down into completely harmless water and oxygen gas.
Which completely explains obligate anaerobes, the bacteria that are actually harmed or killed by the presence of oxygen.
Right. It's not that oxygen magically suffocates them, it's that they completely lack catalase and superoxide dismutase.
So the moment they are exposed to oxygen, those radicals form, and without the enzymes to neutralize them, the bacteria are shredded from the inside out.
That is the exact biochemical mechanism.
Wow.
And beyond just the strict aerobes and anaerobes, you also have to account for microaerophiles.
Meaning they prefer a lower oxygen concentration.
Yeah, lower than what is found in standard room air, usually around 5 to 10% oxygen.
And then you have capnophiles.
Right. Capnophiles require an environment heavily enriched with carbon dioxide compared to regular air.
Okay, so we've set the stage. We have our agar plate. The temperature is 37 degrees. The pH is perfectly buffered at 7.0. The oxygen tension is dialed in.
Now we introduce the bacteria.
Let's track the actual growth curve of a bacterial population in a closed system.
The growth curve is a fundamental microbiological concept, and it's divided into 4 distinct phases.
Starting with the lag phase.
Right. If you look at a graph of this curve, the line is totally flat during the lag phase. The number of cells isn't increasing yet.
But microscopically, there's massive activity going on.
Exactly. This is the period of intense enzyme synthesis and cell elongation.
So they are sensing the new nutrients in the media, turning on specific genetic operons to digest them, and physically growing larger.
They are essentially unpacking their bags, surveying the room, and building the specific tools they need to eat the food we provided.
I love that. And once those tools are built, they enter the log phase.
Yeah. Or logarithmic phase, yes. This is the period of active explosive reproduction via binary fission.
So the population is doubling at a constant exponential rate.
Yep. This is when the bacteria are their healthiest and most metabolically active.
But because it's a closed system, like an agar plate or a broth tube, that explosive growth cannot last forever.
No, it inevitably hits the stationary phase. This plateau occurs due to two simultaneous factors.
Exhaustion of nutrients and accumulation of waste.
Exactly. The total exhaustion of available nutrients and the toxic accumulation of metabolic waste products. The environment becomes harsh.
You see a drop in pH as acidic byproducts build up.
Right. And the defining characteristic of the stationary phase is equilibrium. The number of dead cells exactly equals the number of new cells being produced.
It's a biological stalemate.
And finally, that stalemate breaks, leading into the death phase.
Because the environment is now so severely depleted of nutrients and so utterly toxic, right?
Yeah, the rate of cell death vastly exceeds the rate of new cell division, and the population just crashes.
All right, let's put the listener in the hot seat. It's time for our first board drill.
We are going to present 3 clinical scenarios based on what we just uncovered.
Think through the mechanisms before we reveal the answer.
Okay, scenario 1 for you. You are reviewing the growth dynamics of a fastidious pathogen in a liquid broth culture. You take a spectrophotometer reading and note that the total population number is completely flat and unchanging, yet biochemical assays show a massive spike in intracellular RNA transcription and protein synthesis.
Okay.
Which phase of the bacterial growth curve are you observing? Is it A, lag phase, B, log phase, or C, stationary phase?
Well, if the population is flat, it can't be log phase.
Right.
And if they are actively synthesizing proteins and RNA, they aren't dying or stalemated in the stationary phase either. They are building their tools.
Oh.
The correct answer is A, the lag phase.
Exactly. The key to this question is recognizing the disconnect between population size and metabolic activity.
Because distractor B, log phase, would show a massive spike in the actual number of cells.
Yep. And distractor C, the stationary phase, is incorrect because while the population graph is flat, the cells are starving and dying, not ramping up massive initial enzyme synthesis.
Perfect. Let's run scenario 2.
You isolate a strictly aerobic gram-negative rod from a patient's sputum. You know this organism utilizes oxygen as its final electron acceptor.
Right.
To survive the highly toxic byproducts of this metabolism, the organism must produce an enzyme that specifically neutralizes superoxide radicals. Which enzyme serves this primary protective function? Is it A, coagulase, B, superoxide dismutase, or C, beta-lactamase?
The toxic workplace analogy strikes again.
It does.
We need the first responder HR enzyme. Coagulase builds clots. Beta-lactamase destroys antibiotics. So the enzyme that neutralizes superoxide radicals is B, superoxide dismutase.
You got it.
Oh.
The nomenclature gives it away, but you must know the alternate mechanisms to rule out the distractors confidently on a board exam.
Like distractor A, coagulase, which is a virulence factor that causes blood plasma to clot.
Right, forming a protective fibrin shield around pathogens like Staphylococcus aureus.
And distractor C, beta-lactamase, is an enzyme that chemically cleaves the beta-lactam ring of penicillin-class antibiotics, conferring drug resistance.
Neither has anything to do with oxygen toxicity.
Final scenario for the growth section.
You receive a gastric biopsy from a patient with a suspected peptic ulcer. You are attempting to culture Helicobacter pylori.
Okay.
The standard operating procedure dictates that this organism requires an atmospheric environment containing 10% carbon dioxide, which is significantly higher than ambient room air.
Right.
This specific atmospheric requirement classifies the organism as A, halophile, B, microaerophile, or C, capnophile.
Well, we know halophiles love salt, like our raw oyster vibrio.
Exactly.
Microaerophiles want a tiny sip of oxygen. But an organism craving a high carbon dioxide environment, that is absolutely C, a capnophile.
The clinical reasoning is solid. Distractor A is eliminated because halophiles require high osmotic pressure from salt.
And distractor B, microaerophile, is a common trap.
Right. It is. While many capnophiles are also microaerophilic, the specific requirement for elevated CO2 defines a capnophile. You must match the specific defining trait to the term.
Awesome. So we now have a deep understanding of exactly how bacteria grow and what precise chemical and environmental triggers they need to thrive.
But in a clinical lab or a hospital setting, our primary goal is often the exact opposite.
Right. How do we stop them from growing? Or better yet, how do we completely obliterate them from our surgical instruments and media preparations?
That brings us to section 2, media and sterilization. To understand how to kill them, we first have to understand the media we use to manipulate them.
The absolute foundation of solid culture media is agar.
Agar is a unique polysaccharide derived from marine red algae. It has no nutritional value to the bacteria. It is purely a structural scaffold.
And the physical properties of agar are highly testable, aren't they?
Extremely testable. It melts into a liquid at 100 degrees Celsius, but it remains liquid as it cools, not solidifying until it hits approximately 45 degrees Celsius.
That delayed solidification is incredibly useful. It allows us to cool the agar enough to add heat-sensitive ingredients like sheep blood or antibiotics without destroying them, but before the agar hardens into a gel.
Exactly. Now, most of the media we use is considered complex media, which is a rich soup made up of peptones, beef extracts, and yeast.
But when we are trying to grow obligate anaerobes, the ones whose cells are shredded by oxygen, we have to use specialized anaerobic media.
Right. Anaerobic media is an exercise in chemical engineering. It must contain reducing agents.
And the text specifically highlights two highly testable reducing agents, thioglycolate and cysteine.
These chemicals actively scavenge and bind with any dissolved oxygen in the liquid media, effectively neutralizing it and removing it from the environment.
And there is a crucial procedural step here too. Anaerobic broth tubes should always be heated, usually in a boiling water bath, prior to use.
Yes, this physical heat drives out any trapped, dissolved oxygen gas, resetting the anaerobic environment before you introduce the patient sample.
But the most critical conceptual distinction in this entire area, and a massive trap for board examinees, is the fundamental difference between sterilization and disinfection.
People use these words interchangeably in normal life, but in the lab, conflating them will fail you.
It is an absolute binary. Sterilization means you are actively killing and destroying all microscopic life.
All of it. This is absolute. It includes the highly resistant, dormant bacterial endospores, and it includes all viruses.
If an item is sterile, the microbial count is mathematically zero.
Exactly. Disinfection, on the other hand, is merely a reduction. It inactivates or inhibits pathogenic microorganisms on inanimate surfaces.
But it explicitly may not destroy tough bacterial spores.
Right. Disinfection makes something safer. Sterilization makes something absolute.
Let's run through the physical techniques we use to achieve these states. Number one, the heavy artillery of the laboratory, the autoclave.
The autoclave uses moist heat, specifically steam under pressure. And there's a massive test alert box right here in the text detailing the exact parameters.
Let's hear them.
Well, the physics of the autoclave are what make it sterilize. At standard atmospheric pressure, water boils at 100 degrees Celsius, and the resulting steam is also 100 degrees.
Which is not hot enough to kill endospores.
Exactly. To get steam hotter, you must put it under pressure. The highly testable mandatory parameters you must memorize for a standard autoclave cycle are 15 pounds of pressure per square inch, which allows the steam to reach 121 degrees Celsius, held for a duration of 15 minutes.
15 psi, 121 degrees, 15 minutes. It's a holy trinity of laboratory science.
It really is.
But how do we actually prove the autoclave work? We can't just trust the digital gauges, right? We need biological quality control.
Yes, we use the toughest organisms on the planet to prove our machines work. For the autoclave, the biological QC organisms are the spores of Bacillus stereothermophilus or Bacillus subtilis.
Because these are incredibly resilient, heat-loving, spore-forming organisms.
Right, we place a vial of them into the autoclave run.
Yeah.
If the 121-degree cycle effectively kills these highly resistant spores, which we confirm by trying to culture the vial afterward and seeing no growth, we can be clinically confident that every other vegetative pathogen in that load was completely obliterated.
Makes total sense. That's moist heat. What about dry heat?
Dry heat methods include passing a loop through a Bunsen burner flame, using a benchtop incinerator, or baking glassware in a hot air oven.
Because water conducts heat far more efficiently than dry air, achieving sterilization in a hot air oven requires significantly more extreme parameters than the autoclave.
Oh, absolutely. The standard for a hot air oven is 170 degrees Celsius for 2 full hours.
And then there's pasteurization. The text notes ultra-high temperatures 140 degrees Celsius for just 3 seconds. That sounds intense.
It is, but we need to make a really forceful point about this. The boards will absolutely try to trick you with this.
The text explicitly states it. Pasteurization is not sterilization.
Right. The entire goal of pasteurization, developed by Louis Pasteur, is to heat a liquid like milk or juice just enough to kill specific vegetative pathogens like Salmonella or Listeria.
And to reduce the overall microbial load to prevent rapid spoiling.
Exactly. But it absolutely does not destroy resilient bacterial spores. A pasteurized liquid is not a sterile liquid.
Let me push back on a practical level here. If the autoclave is so incredibly efficient at killing absolutely everything, why do we bother with anything else?
Good question.
I mean, why don't we just blast every single liquid, media, and chemical with steam at 121 degrees for 15 minutes?
Because the sheer violence of that heat denatures proteins and breaks complex chemical bonds.
Oh.
Yeah, if you are preparing a highly sensitive liquid like a carbohydrate solution used for a biochemical fermentation test, a urea broth, or most importantly, a vaccine containing delicate antigens, the intense heat of the autoclave will completely destroy the structural integrity of those molecules.
So you'd end up with a perfectly sterile but completely useless solution. Okay, so you can't boil a vaccine. So how do we sterilize those delicate liquids?
We use mechanical filtration. You pass the sensitive liquid through a physical membrane filter.
And the standard, highly testable pore size for sterilizing filters is 0.22 micrometers to 0.45 micrometers, right?
Yes. Because the vast majority of bacteria are larger than 0.5 micrometers, they are physically trapped on the filter membrane while the sterile liquid passes through into the collection vessel below.
So it achieves sterilization without ever applying destructive heat.
It's pure physical separation.
That makes perfect sense. The text also briefly touches on a few other methods of microbial inhibition, like refrigeration, which simply slows the metabolic rate of the organisms, stalling them in the lag phase.
Right. And desiccation or lyophilization, which is essentially extreme freeze drying.
And it is crucial to note that with desiccation, there's no bacterial multiplication occurring, but the organisms often remain completely viable, right?
Oh yeah, they are in a state of suspended animation, waiting for water to be reintroduced. Lyophilization is actually how we preserve reference strains in the lab for years.
Wow. Then there's osmotic pressure, creating a hypertonic environment to draw water out of the cells, causing plasmolysis.
Which is the fundamental science behind salt-cured meats and high-sugar jams.
Right. And radiation, specifically gamma or UV, which blasts the cells and forms hydroxyl radicals that physically shatter the bacterial DNA backbone.
Yep. And finally, chemical disinfection. Phenolic compounds work by disrupting the lipid bilayer of the cytoplasmic membrane and denaturing internal proteins.
While halogens like iodine and chlorine bleach act as massive oxidizers, right?
Exactly, stealing electrons and destroying cellular machinery.
Let's test that foundational knowledge. Listener, you're back in the hot seat. 3 clinical scenarios on media and sterilization.
Scenario 1. You are the lead technologist in charge of media preparation. You need to sterilize a large batch of standard tryptic soy broth. To achieve true sterilization using moist heat under pressure, you must set the autoclave to which of the following standard parameters. A, 170 degrees Celsius for 2 hours, B, 140 degrees Celsius for 3 seconds, or C, 15 psi at 121 degrees Celsius for 15 minutes.
If we're talking moist heat and true sterilization, we need the magic trinity. 15 pounds of pressure, 121 degrees, 15 minutes. The answer is C.
The rationale here is identifying the specific physical states. Distractor A, 170 degrees for 2 hours, describes the parameters for a hot air oven, which utilizes dry heat, not moist steam.
And distractor B, 140 degrees for 3 seconds, describes ultra-high temperature pasteurization.
Which, as we emphasized, is a process of disinfection and microbial reduction, but explicitly not sterilization.
Let's hear scenario 2.
You are tasked with preparing a highly concentrated urea broth for biochemical testing. You know that urea is a thermolabile molecule. It will completely break down into ammonia and carbon dioxide if subjected to high heat. Which sterilization technique must you utilize to prepare this media safely? A, autoclave, B, filtration, or C, incineration?
Well, we can't boil it or we destroy the chemical we are trying to test. We have to separate the bacteria physically without heat.
So,
The answer is B, filtration through a 0.22 micron membrane.
perfect clinical reasoning. It revolves around the preservation of delicate molecular structures. Both distractor A, the autoclave, and distractor C, incineration, utilize extreme heat that will actively destroy the urea molecules through thermal denaturation.
So filtration is the only method listed that removes the microorganisms without applying destructive thermal energy.
Exactly.
Final scenario for this section.
The laboratory manager is auditing your autoclave logs to ensure the equipment is legally compliant and effectively achieving sterilization. You must provide the biological indicator records.
Right.
Which specific organism is utilized universally as the quality control standard to prove autoclave efficacy? A. Bacillus stereothermophilus, B. Staphylococcus aureus, or C. Pseudomonas aeruginosa.
We need the toughest bug on the block, the heat-loving spore former. That is A. Bacillus stereothermophilus.
Yes. Identifying the biological extreme is the key. You must use an endospore to prove sterilization, and Bacillus is the classic spore-forming genus.
Distractors B and C, Staphylococcus aureus and Pseudomonas aeruginosa, are vegetative bacteria. They are easily killed by heat.
However, they are highly relevant distractors because they are the mandatory quality control organisms used to validate Kirby-Bauer antibiotic susceptibility testing.
Which we will cover shortly. That is a perfect segue. Because once we've successfully cultured the pathogen that we intentionally didn't sterilize, we have to help the physician treat the patient.
Right.
How do these life-saving drugs actually work on a molecular level? We are diving into Section 3, Antibiotics.
To understand antibiotics, we have to start with the core terminology. The first concept is the spectrum of activity.
So narrow spectrum versus broad spectrum.
Exactly. A narrow-spectrum antibiotic acts like a sniper rifle. It only covers certain highly specific groups of bacteria, for example, a drug that only kills gram-positive Kochia but leaves gram-negative rods completely untouched.
While a broad-spectrum antibiotic acts more like a shotgun, it covers a wide swath of both gram-positive and gram-negative organisms simultaneously.
And the fundamental magic of all these drugs relies on the concept of selective toxicity.
Meaning, the antibiotic targets a highly specific structural component or metabolic pathway that the microbe possesses, but that the human host completely lacks.
Right, because if a drug targeted human structures, it wouldn't be an antibiotic, it would just be poison.
So, for example, humans do not have cell walls. Therefore, a drug that specifically attacks the synthesis of a peptidoglycan cell wall will cause massive damage to the bacteria, but simply float past human cells without interacting.
Exactly. Next, we differentiate how the drugs ultimately affect the bacterial population. Bactericidal action means the drug actively and directly kills the bacteria.
It causes lethal structural damage without needing any help from the host's immune system.
Yep. Bactericidal drugs are the heavy hitters you need for a patient with a compromised immune system.
In contrast, bacteriostatic action means the drug merely arrests the growth of the bacteria.
It reversibly inhibits their replication, trapping them in the stationary phase. The drug itself doesn't kill them.
The ultimate destruction of the stalled pathogen depends entirely on the host's own immune defenses, macrophages and neutrophils stepping in to clean up the mess while the bacteria are paralyzed.
Right. Now, what happens in a severe infection when we use more than one drug at a time? Let's break down the mathematical analogies of drug interactions because the text highlights this specifically.
Okay, let's do it. When a physician prescribes a combination of drugs, they are hoping for synergism.
The mathematical analogy the boards love to use is 1 + 2 = 4, meaning the combined concerted effect of the two drugs working together is significantly greater than the simple additive sum of their individual effects.
They physically aid each other. Give me a clinical example of how 1 + 2 = 4.
A classic synergistic combination is a penicillin paired with an aminoglycoside. The penicillin acts first, attacking the bacterial cell wall and creating physical holes in the armor.
Okay.
Normally, the aminoglycoside has a hard time penetrating a thick cell wall. But with the penicillin punching holes in it, massive amounts of the aminoglycoside flood into the cell, bind to the ribosomes, and shut down protein synthesis.
Oh, wow. They help each other work vastly better.
Exactly.
But the terrifying opposite of that is antagonism. The math here is 1 + 2 = 1.
Right. This occurs when one drug actively interferes with the mechanism of the other.
For instance, penicillin only kills bacteria that are actively dividing and building new cell walls.
Yep. So if you simultaneously give a bacteriostatic drug that completely stops the bacteria from dividing, the penicillin suddenly has no target.
The static drug neutralized the cidal drug. You never want antagonism.
Never. Let's move to the mechanics. The antibiotic action table in the text is dense, but it is pure high-yield memorization. Let's group them by their cellular targets, starting with the cell wall synthesis inhibitors.
The largest, most famous group here are the beta-lactams. This class includes the penicillins, the cephalosporins, the carbapenems like imipenem, and the monobactams like aztreonam.
And they all share a chemical structure called a beta-lactam ring.
How does that ring work?
That ring acts as a molecular mimic. It mimics the dealanine peptide chains that bacteria use to cross-link their cell walls.
So when the bacterial enzymes called penicillin-binding proteins grab the antibiotic instead of their actual building materials, the enzyme is permanently locked and disabled.
Exactly. The cell wall falls apart, and the bacterium bursts due to internal osmotic pressure.
But there is a massive clinical note for this entire class. You have to constantly monitor for methicillin-resistant Staphylococcus aureus, or MRSA.
Right, because MRSA has mutated its penicillin-binding proteins so that the beta-lactam drugs can no longer physically fit into the receptor site.
They changed the locks.
They did.
And when a pathogen changes the locks, like MRSA, we bring in the other major cell wall inhibitors, the glycopeptides.
Specifically vancomycin. Vancomycin doesn't bind to the enzyme, it binds directly to the building blocks themselves.
Acting like a giant chemical cap that prevents the wall from being built.
Yeah. Vancomycin is the classic, heavily tested drug of choice for treating severe, life-threatening Clostridium difficile infections in the gut and for treating systemic MRSA infections.
Right, moving inside the cell, past the wall, we have the protein synthesis inhibitors. These drugs target the bacterial ribosomes, which are the factories that translate mRNA into functional proteins.
And bacterial ribosomes are structured differently than human ribosomes. Specifically, they have a 30S and a 50S subunit.
Which provides that selective toxicity we talked about.
Right. Let's start with the drugs targeting the 30S subunit. First are the aminoglycosides. This group includes gentamicin, tobramycin, and amikacin.
They bind the 30S subunit and cause the ribosome to misread the genetic code, creating garbage proteins that kill the cell.
And here's a crucial, highly testable fact. Aminoglycosides are not active against strict anaerobes.
Wait, why not? Why would oxygen matter to a drug?
Because the physical transport mechanism that pulls the aminoglycoside molecule through the bacterial membrane and into the cell requires energy derived specifically from oxidative phosphorylation.
It requires oxygen to power the pump.
Exactly. Without oxygen, the drug simply cannot get inside.
That is a phenomenal connection. Next up on the 30S subunit, the tetracyclines.
Tetracycline and doxycycline. They block the docking site for new amino acids.
The major clinical note here is their severe side effect profile, right? Tetracyclines aggressively bind to calcium ions.
Yes. If given to children or pregnant women, they will deposit in developing bone and teeth, causing permanent severe discoloration and skeletal defects.
And they also wipe out normal flora, frequently leading to superinfections like oral thrush or vaginal yeast.
Yep. Moving to the 50S subunit, we have chloramphenicol.
Chloramphenicol is a powerful 50S inhibitor, but the critical, highly testable toxicity associated with this drug is that it can cross over and suppress human mitochondrial ribosomes in the bone marrow,
which can cause aplastic anemia, a catastrophic and often fatal failure of the bone marrow to produce new blood cells.
So because of this extreme toxicity, its use is heavily restricted to dire, life-threatening scenarios.
Exactly. And the last major group of 50S protein synthesis inhibitors, the macrolides.
This class includes erythromycin and clindamycin.
A key clinical note for clindamycin is its excellent broad-spectrum activity against both gram-positive and gram-negative obligate anaerobes.
So if you have a deep tissue anaerobic infection, clindamycin is often a primary weapon.
It is. So we stop the walls and we stop the proteins. Now let's look at the drugs that stop DNA replication. The nucleic acid synthesis inhibitors.
The main class the boards focus on here are the quinolones, which include ciprofloxacin and norfloxacin.
They work by binding and jamming an enzyme called DNA gyrase, which is responsible for uncoiling the bacterial DNA supercoil.
If the DNA can't uncoil, it can't be replicated, and the cell dies.
Right. These are heavily relied upon for treating tough Pseudomonas aeruginosa infections.
Finally, we have the metabolic pathway inhibitors, specifically the PBA analogs.
The sulfa drugs, or sulfonamides, like sulfamethoxazole, bacteria must synthesize their own folic acid from scratch to survive, and a chemical called PBA is a key building block in that process.
But humans don't synthesize folic acid. We just eat it in our diet, so we lack this pathway entirely.
Exactly. Sulfa drugs are chemical mimics of PBA. They act as a false intermediate.
The bacterial enzyme grabs the sulfa drug instead of PBA, The entire folic acid assembly line shuts down, and the bacteria eventually starves.
Yep, they are very commonly used for uncomplicated urinary tract infection.
Okay, that was a dense, highly technical block. Let's lock those mechanisms in. Listener, back to the hot seat. 3 clinical scenarios on antibiotics.
Scenario 1. You are reviewing the chart of a pediatric patient with a severe, unusual systemic infection. The infectious disease physician wants to prescribe a powerful antibiotic that acts by inhibiting the 50S ribosomal subunit.
Okay.
However, the pharmacist intervenes, warning that this specific drug carries a black box warning for potentially inducing catastrophic aplastic anemia. Which antibiotic is at the center of this consult? A, gentamicin, B, tetracycline, or C, chloramphenicol.
The 50S subunit and the dreaded aplastic anemia toxicity, that specific combination points directly to C, chloramphenicol.
Right. The ability to link the mechanism to the toxicity is what the boards are testing. Distractor A, gentamicin, is an aminoglycoside. It acts on the 30S subunit, not the 50S.
And its classic toxicities are nephrotoxicity and ototoxicity, damage to the kidneys and ears.
Right. Distractor B, tetracycline, is also incorrect. It is a 30S inhibitor, and its classic pediatric side effect is the binding of calcium, leading to permanent discoloration of developing teeth.
Let's hear scenario 2.
A physician orders a 2-drug regimen for a complex infection. Upon reviewing the literature, you note that when used simultaneously, drug A completely arrests the cellular division of the bacteria, which inadvertently prevents drug B from finding the actively synthesizing cell wall targets it requires to cause lethal lysis.
Ouch.
In mathematical terms, the net efficacy of the drugs is defined as 1 + 2 = 1. This physiological interaction describes drug. A, synergism, B, antagonism, or C, reversible inhibition.
One drug is literally stopping the other from working. The math is 1 + 2 = 1. That is a textbook case of B, antagonism.
Recognizing the failure cascade is critical. Distractor A, 1 + 2 = 4, is the definition of drug synergism, where the combination creates a mutually beneficial amplified effect.
Like our penicillin and aminoglycoside example.
Right. Distractor C, reversible inhibition, is a distractor term. It defines the mechanism of a bacteriostatic action itself, but does not describe the interaction between 2 different drugs.
Final antibiotic scenario.
A patient is admitted to the ICU with a severe necrotic skin infection. Cultures confirm the presence of methicillin-resistant Staphylococcus aureus, or MRSA.
Okay.
The standard beta-lactam class is completely ineffective because the organism has mutated its penicillin-binding proteins. The physician immediately orders the gold standard intravenous drug for MRSA, which works by physically capping the D-alanine peptide precursors.
Yeah.
This drug belongs to which structural class of antibiotics? A, beta-lactams, B, glycopeptides, or C, macrolides.
The drug they're ordering is vancomycin, and vancomycin belongs to the glycopeptide class. Answer B.
Correct. You must know the drug and its broader chemical family. Distractor A is incorrect because the beta-lactam class is exactly what the MRSA is resistant to.
Right. It includes the penicillins, cephalosporins, and carbapenems.
And distractor C is incorrect because macrolides, like erythromycin, are 50S protein synthesis inhibitors, not massive cell wall capping agents like the glycopeptides.
Outstanding. So it's great to know how these drugs function in theory on a molecular level, but we don't treat patients with theory.
No, we don't.
We have to definitively prove that a specific drug will actually kill the specific pathogen we just isolated from the patient's blood. We have to run the tests.
This brings us to Section 4, susceptibility testing. The foundation of all manual susceptibility testing is the Kirby-Bauer disc diffusion method.
It seems simple. You swab bacteria on a plate, drop paper discs soaked in antibiotics on it, and measure the clear circles where the bugs died.
But the physics of how that drug diffuses through the agar jelly is incredibly complex.
Because of this, the boards love to test the strict standardized parameters required for this test to be legally and medically valid.
Right. If you change one variable, you change the physical diffusion rate of the drug, and you generate a false result.
Every single variable is locked down by regulatory standards. Let's lay out the absolute rules.
The agar must be Mueller-Hinton agar. It is formulated specifically because it does not contain inhibitors that interfere with sulfonamides or tetracyclines.
And it must be poured to a uniform, mathematically exact depth of 4 millimeters.
The pH of the cooled agar must be strictly maintained between 7.2 and 7.4. It must contain physiologic concentrations of calcium and magnesium cations.
And the plates are incubated at 35 degrees Celsius in ambient air, not CO2, right?
Yes, because CO2 lowers the pH of the agar.
And what about the actual bacterial suspension we swab onto that perfectly engineered plate?
The inoculum density is just as critical. You cannot just guess. It must be standardized to match a McFarland 0.5 turbidity standard.
Visually, it looks like very slightly cloudy water.
Mathematically, it corresponds to roughly 1.5 times 10 to the 8th organisms per milliliter. If you have too many bugs or too few bugs, the battle against the antibiotic disc is fundamentally unfair.
Finally, quality control must be performed rigorously, weekly, and with every single new lot of agar or antibiotic discs.
The standard highly testable QC organisms are Escherichia coli, Staphylococcus aureus, and Pseudomonas aeruginosa.
That's the agar disc diffusion method. The text also covers liquid broth microdilution methods, which provide us with hard numbers.
Yes. You must be able to differentiate the clinical meaning of the MIC and the MBC. The MIC, or minimum inhibitory concentration, is the lowest concentration of the drug that prevents visible in vitro growth.
Visually, if you are looking at a series of broth tubes with decreasing amounts of antibiotic, the MIC is the very first clear tube.
But the key word there is inhibits. It prevents visible growth. But are the bacteria in that clear tube actually dead, or are they just stunned?
That is the vital question, and that is where the MBC comes in.
The MBC, or minimum bacteriostatic concentration, though clinically and accurately it's referred to as the minimum bactericidal concentration, is the lowest concentration of drug that results in greater than 99.9% lethal killing of the initial bacterial inoculum.
To find the MBC, you can't just look at a clear tube. You actually have to take a sample from the clear MIC tubes and subculture it onto a fresh, drug-free agar plate.
Exactly. If the plate grows tomorrow, the drug only stunned them. If the plate remains empty, you have found your bactericidal concentration.
And there's a third highly specialized test mentioned here, the Schlichter test.
The Schlichter test is a fascinating biological assay. Instead of testing standardized factory-made antibiotics against a bug, it is a serum bactericidal assay.
You draw the patient's actual blood, which currently contains the antibiotic they are taking, metabolized by their liver, bound to their unique serum proteins.
And you test their own serum against the pathogen you isolated from them. It is the ultimate real-world test.
It tells the physician if the therapy is actually achieving lethal bactericidal levels in the patient's own bloodstream.
Right.
I love this next part. The sources of error in disc diffusion table. I treat this like a forensic investigation.
It really is.
You look at an abnormal, ruined result on the Kirby-Bauer plate, and you use physics and biology to deduce exactly what procedural error the technologist made. Let's walk through these forensic scenarios.
Scenario 1. You look at your Kirby-Bauer plate, and all the zones of inhibition across every single different drug class are universally way too large. What physical error happened?
If every zone is too large, it means the drugs diffused outward way too fast and too far, where the bacteria put up no fight.
Okay. That means the agar depth was poured too thin, maybe 2 millimeters, instead of the required 4 millimeters, creating less 3D resistance for the drug molecules. Or the technologist made the inoculum too light. There weren't 10 to the 8th bacteria. Maybe there were only 10 to the 5th. There simply weren't enough bacteria to fight back, so the standard amount of drug wiped them out easily.
Exactly.
The physical resistance was altered.
Conversely, what if the zones are universally too small across the entire plate?
It's the exact opposite problem. The agar was poured too thick, maybe 6 millimeters deep, acting like a dense swamp that restricted the drug from diffusing outward.
Or the inoculum was way too heavy, acting like a massive, overwhelming bacterial army that absorbed and neutralized the drug before it could spread.
Perfect physics. Now let's look at chemical errors, specifically the pH. What if the pH of the Mueller-Hinton agar is too low, meaning it's too acidic?
Acidic pH causes specific divergent chemical errors depending on the drug's ionization. Tetracycline and clindamycin zones will be falsely too large in acid, but aminoglycoside zones will be falsely too small.
And if the pH is too high, too alkaline, you see the exact opposite chemical effect.
And what about those specifications we mentioned, calcium and magnesium?
This is a highly specific, very testable interaction. It specifically affects aminoglycoside antibiotics being tested against Pseudomonas aeruginosa.
Because Pseudomonas has pores in its membrane.
Right. Calcium and magnesium ions physically bind to and block those pores. If the cation levels are too high in the agar, the aminoglycoside cannot enter the cell, and the zone of inhibition will be falsely too small.
And if the cations are too low, the pores are wide open, the drug floods in, and the zone is falsely too large.
Excellent breakdown. The final visual error scenario is the alarming zone within a zone phenomenon.
Oh, this one is tricky.
You see a clear, beautiful zone of inhibition around the disc, but when you look closely, there are tiny, distinct colonies growing inside that clear zone right next to the antibiotic. What is the cause?
A few possibilities. It could be a highly motile, swarming Protea species literally swimming into the toxic zone after the drug diffused.
Right.
It could be the feather edges seen around ampicillin discs with certain strains of S. aureus. But the most critical clinical concern is heteroresistance.
Yes. You had a mixed culture, or worse, a tiny subpopulation of the pathogen has genetically mutated to become resistant.
The drug killed the 99% susceptible population, creating the clear zone. But the 1% resistant mutant thrived in the empty space. You must report that isolate as resistant.
And right below this table is a massive test alert. The text explicitly warns you to master 3 specific areas: the preparation standards for Kirby-Bauer plates and inoculum, the interpretation of results, including that entire troubleshooting error table we just forensically analyzed,
And knowing the specific genus and species of the organisms used in Kirby-Bauer quality control.
All right, listener, it is time for the board drill on susceptibility testing. Let's see if you can troubleshoot like a master technologist.
Scenario 1. A junior technologist prepares a fresh batch of Mueller-Hinton agar plates, but mistakenly pours them to a depth of 6 millimeters instead of the rigorously standardized 4 millimeters. When these overly thick plates are subsequently utilized for standard Kirby-Bauer disc diffusion testing, what will be the resulting impact on the interpretation? A. The zones of inhibition will be universally too large, resulting in false susceptibility reports. B. The zones of inhibition will be universally too small, resulting in false resistance reports. Or C. The depth alteration will exclusively cause an indeterminate zone around the methicillin disc.
We apply the physics of diffusion. The agar is a swamp. It's too thick, acting like a massive roadblock.
Right.
The drug molecules physically cannot diffuse as far outward as they're supposed to. Therefore, the circles of death will be small. The answer is B, zones universally too small.
Your physical deduction is flawless.
Yeah.
Distractor A, zones universally too large, would manifest if the agar was poured far too thin, perhaps 2 millimeters, allowing the antibiotic to diffuse outward with abnormally low resistance.
And distractor C is a hyperspecific issue related to temperature and beta-lactamase degradation, not a general physical agar depth error that impacts every disc on the plate.
Let's run scenario 2.
You are interpreting a Kirby-Bauer plate for a highly virulent, spot-on biochemical reasoning. Let's systematically dismantle the distractors. Distractor B, a highly acidic pH, would actually cause the aminoglycoside molecule to ionize poorly, resulting in a falsely small zone, not a large one.
And distractor C, an overly heavy inoculum of McFarland 1.0, would overwhelm the drug and cause all zones across the entire plate to be universally too small.
Final susceptibility scenario.
Infectious disease specialist calls the laboratory, inquiring about a highly resilient enterococcal endocarditis case. They need to know the absolute lowest concentration of an intravenous drug that prevents visible in vitro replication of the organism so they can dose the patient just above that threshold.
Okay.
Which specific laboratory value is the physician requesting? A, the minimum bactericidal concentration, or MBC. B, the minimum inhibitory concentration, or MIC. Or C, the Schlichter titer level.
The key phrase in the physician's request is prevents visible in vitro replication, not necessarily killing it, just stopping it.
Right.
That is the exact definition of the minimum inhibitory concentration. Answer B, MIC.
Exactly. Distractor A, the MBC, requires an additional subculture step to definitively prove greater than 99.9% actual killing of the bacteria, which exceeds the physician's request for mere inhibition of visible growth.
And distractor C, the Schlichter test, uses the patient's actual circulating serum rather than standard factory antibiotic dilutions, which is not what the physician asked for.
So before we can even run these elaborate susceptibility tests, we have to actually isolate the pathogen from the messy, complex patient sample.
Which requires choosing the exact right real estate to grow them on the agar media.
This brings us to Section 5, Special Media and Confusing Tests.
The human body is packed with normal, healthy flora. If you swab a throat and put it on a plate that grows everything, you will just get a lawn of normal bacteria, and the pathogen will be completely hidden.
So we rely on highly engineered media. Let's do a rapid, deep dive overview of the routine media. You have your standard blood agar, or BAP, usually made with 5% sheep blood.
It is considered non-selective and enriched. It grows almost everything. But crucially, it is differential because it allows us to visualize hemolytic reactions, the bacteria literally destroying the red blood cells.
Then you have chocolate agar. It isn't made of chocolate. It's essentially blood agar that has been heated until the red blood cells lyse, releasing their internal contents.
Why do we do this? Because fastidious, wimpy organisms like Haemophilus and Neisseria require internal cellular factors, specifically hemin, which is factor X, and NAD, which is factor V, to survive.
But they completely lack the enzymes to break open intact red blood cells themselves. Chocolate agar does the work for them.
Exactly. Then we move to selective media, which contains chemical roadblocks to inhibit unwanted normal flora. PEA, or phenylethyl alcohol agar, disrupts the lipid membranes of gram-negative organisms, effectively paralyzing them.
Therefore, it strictly selects for the growth of gram-positive kochi and anaerobic bacilli.
CNA, Columbia colistin nalidixic acid agar, utilizes two powerful antibiotics to achieve the exact same goal, isolating gram-positives by killing gram-negatives.
Thayer-Martin agar is an incredible piece of selective engineering. It is a chocolate agar base, but it is spiked with a cocktail of antimicrobials. Vancomycin to kill gram-positives, colistin to kill gram-negatives, nystatin to kill yeast, and trimethoprim to kill swarming proteus.
It is designed to be a completely toxic wasteland to everything on Earth, except for pathogenic Neisseria gonorrhea and Neisseria meningitis.
You also have Campylobacter agar, heavily spiked with antibiotics specifically designed to isolate Campylobacter from stool samples.
You have thioglycolate broth, which we discussed earlier as an oxygen-scavenging liquid media used as a universal backup to catch trace amounts of strict anaerobes.
And you have Lowenstein-Jensen, or Middlebrook 7H10 agars. These contain malachite green dye to inhibit normal respiratory flora, and they are the classic, slow-growing agars for isolating Mycobacterium tuberculosis.
But the text goes deeper into the strict anaerobic media. Identifying anaerobes relies heavily on specific pleats.
Like BBE, or Bacteroides bilesculin agar, which is spiked with the gentamicin. It specifically selects for the incredibly robust Bacteroides fragilis group.
If B. fragilis grows, it hydrolyzes the esculin in the agar, turning the surrounding media pitch black.
KVLB, kanamycin vancomycin lake blood agar, is also for isolating Bacteroid species, but it is specifically formulated to enhance their unique dark pigment production.
CCFA, cycloserine cefoxitin fructose agar, is the gold standard selective media for isolating the dangerous gut pathogen Clostridium difficile directly from stool.
But what really fascinates me is the hyperspecific, bizarre diets of some of these rare pathogens. The special media table in the text is just a list of extremely picky eaters. Let's run through them and highlight why they need these weird ingredients.
Absolutely. Bordet Jengu agar is affectionately known as cough plate media. It utilizes potato infusion and blood and is explicitly engineered for isolating Bordetella pertussis, the agent of whooping cough.
Next is BCYE, buffered charcoal yeast extract. This is the classic media for Legionella species. But why the charcoal?
Legionella is incredibly sensitive to toxic oxygen radicals and metabolic byproducts present in standard agar. The charcoal acts as a massive chemical sponge, absorbing and neutralizing those toxins so the Legionella can finally grow.
Cysteine glucose agar is spiked with sulfur-containing amino acids, which is an absolute requirement to culture the highly infectious agent of tularemia, Francisella tularensis.
Fletcher's medium is a semi-solid broth enriched with rabbit serum used to culture the corkscrew-shaped Spirochete leptospira.
Kelly's medium is a complex, highly enriched broth required to cultivate the Lyme disease agent, Borrelia burgdorferi.
Skiro agar is a blood agar base packed with antibiotics to isolate the ulcer-causing Helicobacter pylori from gastric biopsies.
TCBS, thiosulfate citrate bile salts sucrose agar, has an extremely high pH and heavy bile salts. It is a highly selective differential wasteland designed specifically for our salt-loving Vibrio species.
V agar utilizes human blood rather than sheep blood because it is the only way to visualize the unique beta hemolysis of Gardnerella vaginalis.
And finally, Tinsdale or Lafleur's medium, which utilize potassium tellurite, are used to culture and biochemically identify Corynebacterium diphtheria.
And before we leave growth requirements, we have to mention cold enrichment. The text uses a very memorable graphic here to hammer the point home, an igloo and a penguin.
It's an incredibly important biological loophole. Most pathogens, as we said, want to be at a comfortable 37 degrees.
But two specific organisms, Listeria monocytogenes and Yersinia enterocolitica, possess specialized cold shock proteins that allow them to tolerate and even thrive in much colder temperatures.
So if a stool or tissue sample is heavily contaminated with normal flora, you can place the broth in the refrigerator.
The normal flora will completely stop growing and slowly die off, but the Listeria or Yersinia will continue to slowly multiply, enriching their numbers over several days until they're easy to isolate on a plate.
Okay, now we arrive at one of the most historically confusing areas of clinical ID, the confusing test table. These are old eponymous tests named after the physicians who invented them.
I call these the old law firm tests because they all sound like partners at an ancient legal practice. Schick, Dick, Schlichter, Schultz, and Ehrlich. Let's decipher the immunology behind them.
They are easily confused, hence the table. Let's separate them by mechanism. The Schick test is an in vivo test. A tiny amount of diphtheria toxin is injected under the patient's skin.
If the patient does not have neutralizing antibodies, meaning they are susceptible to the disease, the toxin causes a localized inflammatory rash.
Therefore, a positive Schick test detects susceptibility to Corynebacterium diphtheriae.
The Dick test is functionally identical, but for a different disease. It is an in vivo injection of the erythrogenic toxin produced by Streptococcus pyogenes. A localized rash confirms susceptibility to scarlet fever.
We already covered the Schlichter test, that's the in vitro serum bactericidal level, testing the patient's blood against their own pathogen to prove antibiotic efficacy.
Right. Then there is the Schultz-Charlton test. This is an in vivo diagnostic test, not a susceptibility test.
If a patient presents with a full body rash, the physician injects specific Streptococcus antiserum directly into the rash.
If the rash dramatically blanches and fades at the injection site, it proves that the antibodies successfully neutralized the toxin. It confirms the rash is indeed scarlet fever from S. pyogenes.
And finally, the Ehrlich test. This is an in vitro immunodiffusion assay performed on an agar plate.
You strike the isolated strain of Corynebacterium diphtheriae across the plate and lay a paper strip soaked in antitoxin perpendicular to it.
As they both diffuse into the agar, if the bacteria is actively producing the deadly toxin, a white line of precipitation will form exactly where the toxin and antitoxin meet in optimal proportions. It proves the organism is actively lethal.
And we have a test alert here demanding we master this section.
It emphasizes knowing the anaerobic media, specifically BBE for B. fragilis and CCFA for C. difficile.
It demands knowledge of the special media with a bolded emphasis on BCYE for Legionella.
And it insists, the exact purpose of those confusing tests we just outlined.
Let's lock it in. Listener, prepare for the board drill on Section 5.
Scenario 1. A 65-year-old male presents with severe atypical pneumonia following a stay at an older hotel with a centralized cooling tower.
Okay.
Suspecting a specific fastidious intracellular pathogen known to be incredibly sensitive to toxic metabolic byproducts in routine agar, the laboratory strictly utilizes buffered charcoal yeast extract media for isolation. Which organism is the physician attempting to culture? A. Legionella pneumophila, B. Vibrio cholerae, or C. Bordetella pertussis.
A test alert warned you this was coming. The clinical history of a cooling tower, combined with the absolute requirement for the charcoal sponge in BCYE agar, defines A. Legionella pneumophila.
Exactly. Distractor B. Vibrio cholerae is an enteric pathogen that requires the highly alkaline TCBS agar, not BCYE.
Distractor C. Bordetella pertussis is a respiratory pathogen, but it classically requires the potato infusion of Border Jengu cough plates, not charcoal yeast extract.
Scenario 2.
A pediatric patient presents to the clinic with a diffuse erythematous rash. To definitively diagnose the etiology of the rash, the physician performs the Schultz-Charlton test by injecting a highly specific antiserum directly into the affected tissue.
Right.
Within hours, the rash locally blanches and fades at the injection site, indicating toxin neutralization. What organism is fundamentally responsible for the systemic rash? A. Corynebacterium diphtheriae, B. Streptococcus pyogenes, or C. Listeria monocytogenes.
The blanching rash in response to antiserum is the historical hallmark of the Schultz-Charlton diagnostic test for scarlet fever. The organism responsible for the erythrogenic toxin of scarlet fever is B. Streptococcus pyogenes.
The law firm tests strike again. Distractor A, Corynebacterium diphtheria, is associated with the Schick test to determine patient susceptibility and the Ehrlich immunodiffusion test to confirm in vitro toxin production, but it does not cause the blanching rash of scarlet fever.
Distractor C, Listeria, has no associated historical eponymous immune test.
Final scenario for media.
You are processing a foul-smelling deep tissue aspirate from a diabetic foot ulcer. You are utilizing a full battery of strict anaerobic media.
Okay.
You observe luxurious growth on a specialized laked blood agar plate containing kanamycin and vancomycin. Furthermore, the colonies are developing a distinct, dark, brown to black pigment. This specialized media is engineered to specifically isolate and enhance the pigment of which anaerobic genus: A, the Bacteroides fragilis group; B, pigmented Bacteroides and Prevotella species; or C, Clostridium difficile.
The media described as KVLB. While it grows many anaerobes, its specific claim to fame is isolating and enhancing the pigment of the Bacteroides and Prevotella groups. Answer B.
Correct. Distractor A, the Bacteroides fragilis group, is classically isolated on BBE agar, where it hydrolyzes esculin to turn the media black, which is a biochemical reaction, not a true biological pigment enhanced by late blood.
Distractor C, Clostridium difficile, is an enteric pathogen specifically targeted by the selective CCFA media, not KVLB.
Okay, we understand how to grow them, how to select for them, and how to test them. But getting them to grow on a perfectly engineered plate is only half the battle in a hospital.
First, the biological sample has to actually arrive in the laboratory intact, without the pathogen being ruined or overgrown during transit.
This brings us to Section 6, pre-analytical variables, focusing on specimens and blood cultures.
The universal golden rule for all clinical specimen handling is that timing and the transport environment are everything. For example, the text notes the optimal timing for diagnosing a Salmonella typhi infection.
The bacteria migrate. You have the highest probability of culturing the pathogen from the blood during the first week of the fever.
But by the second and third weeks, the bacteria have moved into the gut, and you must culture the stool to find them. If you pull the wrong sample at the wrong time, you miss the diagnosis.
Another absolute universal rule: you must always collect microbiological specimens prior to the initiation of antibiotic therapy.
If the patient is already pumped full of broad-spectrum drugs, the pathogen will be stunned or killed in the collection tube, and our cultures will be sterile even though the patient is still infected.
Once collected, specimens should be transported to the lab and set up on media within 2 hours to prevent the overgrowth of contaminating normal flora.
But the most heavily tested pre-analytical rules, the ones that cause immediate specimen rejection, involve temperature abuse.
There is a massive, bolded rule in the text. Never, under any circumstances, refrigerate spinal fluids, anaerobic specimens, or suspected Neisseria gonorrhea or GC specimens.
Let's explain why. Cerebrospinal fluid pathogens like Neisseria meningitis and Haemophilus influenza, as well as GC and strict anaerobes, are incredibly fragile.
They are exquisitely adapted to the warm, perfectly regulated 37-degree interior of the human body.
When you plunge them into a 4-degree refrigerator, they undergo massive, irreversible thermal shock. Their lipid membranes crystallize and shatter. The cold literally kills them rapidly.
If a CSF specimen does get refrigerated, or if any sample arrives in a leaky container, or on a completely dried-out swab where the bacteria have desiccated, or in an unapproved chemical preservative like formalin, that meets the strict criteria for rejection.
We have a legal and ethical obligation to reject those samples, call the physician, and request a redraw. Because processing a compromised sample will generate a false negative result, which is clinically far more dangerous than no result at all.
Let's shift specifically to blood culture collection, which has its own very strict set of life-saving rules.
The primary concern with drawing blood for culture is preventing contamination with the patient's own normal skin flora, like Staphylococcus epidermidis.
You must prep the venipuncture site strictly, usually with a vigorous dual-step process. Scrubbing with 70% alcohol to strip away the lipids, followed by an iodine or chlorhexidine swab that must be allowed to dry completely to kill the deep pore bacteria.
When is the physiological best time to actually draw that blood?
The ideal time to capture the bacteria swimming in the bloodstream is just prior to a fever spike, which corresponds to the massive influx of organisms from the infected tissue into the blood.
And how many bottles do we draw? Can we just draw one and call it a day?
Absolutely not. The rule stated in the text is to draw at least 2 complete sets of cultures from 2 entirely different venipuncture sites.
This allows us to differentiate true bacteremia from a localized skin contamination. If both arms grow the same bug, it's real. If only one arm grows a skin bug, it's a contaminated draw.
But critically, you should draw no more than 3 sets in a 24-hour period. Drawing more than 3 doesn't statistically increase your diagnostic yield, but it rapidly depletes the patient's blood volume, leading to hospital-acquired anemia.
What if the patient is crashing and they are already on high-dose 5V antibiotics when the physician orders the blood cultures?
Then you must use specialized blood culture bottles. These bottles contain an ARD, an antibiotic removal device, or specially engineered resin beads.
These microscopic beads physically bind to and sequester the antibiotic molecules floating in the blood sample, permanently neutralizing them, which frees the suppressed bacteria to finally grow in the broth.
The text also notes the isolator system, which involves chemically lysing the patient's red and white blood cells to release intracellular pathogens. The text explicitly notes this is considered the best method for isolating stubborn fungi and acid-fast organisms from blood.
And there's a giant test alert box staring at me right here regarding blood volumes.
Yes, it emphasizes the strict, mathematically required dilution rule for blood cultures. You must achieve a 1:10 dilution of whole blood to culture broth inside the bottle.
Why? Because human blood is a lethal weapon. It is packed with complement proteins, active antibodies, lysozymes, and hungry white blood cells.
If you put too much blood in a small amount of broth, the patient's own immune system will continue to fight and kill the bacteria inside the glass bottle.
By achieving a 1:10 dilution, for example, injecting 10 milliliters of adult blood into 90 milliliters of broth, you sufficiently water down and neutralize those inherent antimicrobial factors, giving the bacteria a chance to grow.
All right, time to test those strict pre-analytical rules. Listener, back to the hot seat for Section 6.
Scenario 1. A frantic courier delivers a stat batch of specimens from an outpatient clinic. Due to a massive traffic delay, the courier placed the transport cooler on ice to preserve the samples.
Oh, no.
Which of the following specimens must the laboratory technologist strictly reject and request an immediate redraw for, specifically because the refrigeration will cause irreversible thermal shock to the suspected fastidious pathogen? A midstream clean cache urine, B, a lumbar puncture spinal fluid, or C, an expectorated sputum.
We remember the golden rule of thermal shock. Cold preserves urine and sputum by stopping normal flora from overgrowing. Cold actively murders the fragile pathogens in CSF. The answer is B, spinal fluid.
Correct. The text explicitly dictates that you must never refrigerate spinal fluids, anaerobic transport vials, or suspected GC swabs.
Distractor A, urine, and distractor C, sputum, are routinely refrigerated if laboratory processing is delayed by more than 2 hours, specifically to prevent the rapid overgrowth of contaminating normal flora from ruining the quantitative analysis.
Scenario 2.
A phlebotomist is training a new hire on the critical parameters for drawing blood cultures on an adult patient with suspected sepsis. To effectively neutralize the inherent bactericidal properties of the patient's circulating complement proteins and lysozymes, what is the mathematically required ratio of patient blood to culture broth inside the bottle? A, 1:2, B, 1:5, or C, 1:10.
We have to water down the immune system. The test alert made this explicit. It must be a massive dilution. Answer C, 1:10.
Exactly. A 1:10 ratio is universally required to dilute out the patient's innate antimicrobial factors that would otherwise inhibit bacterial growth in vitro.
Distractors A and B simply do not provide a sufficient volume of broth to adequately dilute those powerful serum factors, leading to false negative cultures.
Final scenario for pre-analytical.
An aggressive intensive care physician is managing a patient with a fever of unknown origin. In an attempt to maximize the diagnostic yield, the physician orders blood cultures to be drawn every 2 hours.
Okay.
According to standard microbiological guidelines, what is the maximum number of blood culture sets that should ideally be drawn from a patient in a single 24-hour period to optimize detection without causing iatrogenic harm? A, 2, B, 3, or C, 4?
We want to catch the bug, but we don't want to bleed the patient dry. The rule is at least 2, but no more than 3. The maximum is B3.
Correct. Statistical studies demonstrate that drawing 2 or 3 sets provides greater than 95% sensitivity for detecting true bacteremia.
Distractor C, 4 sets, is incorrect because drawing more than 3 sets rarely yields any new microbiological information, but significantly contributes to iatrogenic anemia, requiring unnecessary blood transfusions for the patient.
Okay, the specimen was handled perfectly, transported at the correct temperature, accepted by the lab, plated on specialized media, and it's been incubating. We have colonies.
Now let's look at the actual physical and chemical diagnostic tools we use to see what's growing. Section 7. Diagnostics, focusing on microscopy and stains.
We rely on several entirely different physics of microscopy to visualize these organisms. Standard bright field light microscopy has a resolving power of about 0.2 micrometers. When you use the oil immersion lens, you multiply the 10x ocular lens in the eyepiece by the 100x objective lens to achieve a total functional magnification of 1000x.
The oil is critical there, right?
Yeah.
It bridges the gap between the glass slide and the lens, preventing the light from bending and scattering into the air, driving all the photons straight into your eye.
Then there is dark field microscopy. Dark field utilizes a special condenser that blocks direct light. It uses only reflected light hitting the specimen from oblique angles.
The background appears pitch black, and the organisms appear brightly illuminated, like dust motes glowing in a sunbeam.
This is the classic, heavily tested method required for visualizing spirochetes like Treponema pallidum, the causative agent of syphilis. They are so incredibly thin, often less than 0.15 micrometers wide, that they are completely invisible with a standard bright field gram stain.
We also use fluorescence microscopy, which operates in the near UV range, blasting the sample with high-energy light and catching the lower-energy visible light that the fluorescent dyes emit.
And finally, electron microscopy, which uses a beam of electrons instead of light. It has an incredible resolving power down to 0.001 micrometers, making it absolutely essential for visualizing the intricate structures of viruses.
Now, earlier we mentioned anaerobic incubation. Let's detail the exact atmospheric chemistry occurring inside those anaerobic jars or chambers. The text is very specific and the boards will ask for the recipe. It requires an exact gas mixture of 10% hydrogen, 5% carbon dioxide, and 85% nitrogen.
But the gas alone isn't enough. It absolutely requires palladium crystals to act as a chemical catalyst.
The palladium drives a room temperature reaction that violently binds the 10% hydrogen with any residual toxic oxygen in the chamber to form harmless water condensation.
10% each, 5% CO2, 85% N2, and a palladium catalyst. Got it. Okay, here is the absolute core of Section 7, and frankly, the core of clinical microbiology, the stains table. I'm going to break down the chemistry of these stains step by step. First, the undisputed king of the lab, the gram stain.
Walk us through the specific reagents and their mechanisms.
Step 1. The primary stain is crystal violet. This is a positively charged dye that floods the cells and binds to the negatively charged bacterial components. Everyone turns purple.
Step 2. We add iodine. The iodine acts as a chemical mordant. It doesn't change the color. It physically binds with the crystal violet inside the cell wall, forming massive, bulky, crystal violet iodine molecular complexes.
Step 3. The decolorizer, a mixture of alcohol and acetone. This is the critical defining step.
And explain the physical mechanism of that decolorizer on the cell walls.
Gram-positive bacteria have a massively thick layer of peptidoglycan. The alcohol dehydrates this thick layer, causing the microscopic pores in the wall to shrink and tighten. Those massive, bulky iodine dye complexes are physically trapped inside. They stay purple.
Gram-negative bacteria have a very thin layer of peptidoglycan and a thick outer lipid membrane. The alcohol rapidly dissolves that lipid membrane and blasts right through the thin peptidoglycan, washing all the dye out. They become completely colorless.
Step 4. We add the counterstain, safranin, a pink dye. The trapped gram-positives are already dark purple, so they ignore it. The empty gram-negatives soak up the pink.
Beautifully explained. Now let's move to the specialized stains used specifically for mycobacteria, the acid-fast bacilli responsible for tuberculosis.
Their cell walls are composed of up to 60% complex lipids, specifically mycolic acids, which act like a dense coat of biological wax. A standard gram stain cannot penetrate this wax.
The traditional method is the Kinyon, or Zeal-Nielsen stain.
Right. The primary stain here is carbolfucin, a highly concentrated, lipid-soluble red dye. You literally have to force it into the wax. The decolorizer is highly aggressive, acid alcohol, and the counterstain is methylene blue.
The interpretation here hinges on that wax coat. Because the mycobacteria have that dense mycolic acid layer, once the carbolfucin is forced inside, the wax resists decolorization, even by the aggressive acid alcohol. They hold fast to the acid.
So if they retain the primary carbolfucin, they stain hot pink, and we call them acid-fast positive. Background cellular debris, human tissue, and all non-acid-fast bacteria are stripped of the red dye and take up the methylene blue, staining entirely blue.
But there is a much faster, more sensitive fluorescent method for screening mycobacteria, the auramine-rhodamine stain.
Yes. The primary stain is a highly fluorescent mixture of auramine and rhodamine dyes that specifically bind to the mycolic acids. The decolorizer is still the aggressive acid alcohol. But the counterstain is a unique chemical, potassium permanganate.
Potassium permanganate. Why use a heavy metal compound instead of a normal dye?
Because it acts as a powerful quenching agent, it eliminates all non-specific background fluorescence from the human tissue and debris.
If mycobacteria are present, they will retain the primary fluorescent dyes and appear as blazing, bright orange fluorescing rods standing out sharply against a pitch-dark background. It makes scanning slides infinitely faster.
Finally, the stain for visualizing yeast and fungi directly in clinical specimens, chalcofluor white.
The reagent combination here is chalcofluor white dye mixed with a 10% solution of potassium hydroxide, or KOH. The 10% KOH is incredibly caustic. It is not a stain.
Its specific vital purpose is to dissolve, digest, and completely break down background human cellular debris, keratin, and thick mucus, clearing the visual field.
Once the field is cleared, the chalcofluor white selectively binds directly to the tough chitin and cellulose in the fungal cell walls. Under fluorescent UV light, positive yeast or fungal hyphae will exhibit a brilliant, glowing, bluish-white fluorescence.
That is a phenomenal amount of reagent chemistry. Let's see if the listener retained the mechanisms. Board drill for Section 7.
Scenario 1. You are validating a new automated stainer for the Microbiology department. You are programming the sequence for the highly sensitive fluorescent oromine-rhodamine stain procedure to effectively quench the autofluorescence of the background human tissue and allow the acid-fast bacilli to shine clearly. Which specific chemical must you load as the final counterstain? A. Methylene blue, B. Potassium permanganate, or C. Safranin?
We just highlighted the need for a heavy metal quenching agent to darken the background. That is B, potassium permanganate.
Correct. The distractors represent the counterstains for entirely different diagnostic protocols. Distractor A, methylene blue, is the standard counterstain used in the traditional non-fluorescent Kinyan or Ziehl-Neelsen acid-fast stain.
Distractor C, safranin, is the pink counterstain used in the fundamental Gram stain procedure.
Scenario 2.
A dermatologist sends a thick skin scraping to the laboratory, requesting a direct microscopic examination for suspected ringworm, a dermatophyte fungus. The standard operating procedure dictates a preparation utilizing chalcofluor white. What is the primary biochemical purpose of adding 10% KOH to this specific microscopic preparation? A, to act as a chemical mordant, cross-linking the dye to the chitin. B, to aggressively decolorize the primary stain from non-fungal elements. Or C, to dissolve and break down the obscuring background keratin, debris, and mucus.
KOH is highly caustic lye. It doesn't stain, it digests. The clearing agent is C, to break down debris and mucus.
Exactly. You must know the function, not just the name. Distractor A describes the precise function of iodine in the Gram stain procedure, creating massive dye complexes. Distractor B describes the function of the alcohol-acetone mixture, or acid-alcohol, in their respective staining protocols, washing dye out of susceptible cells.
KOH is strictly for digestion and clearing of human tissue.
Final scenario for diagnostics.
You are inspecting the biological indicators for the laboratory's primary anaerobic chamber. The indicators show unacceptable trace oxidation, meaning the chamber is failing to maintain strict anaerobiosis. To reset the atmospheric environment correctly, you must verify that the incoming gas tanks contain which specific mathematically required gas mixture. A, 5 to 10% CO2 with the remainder as ambient air. B, 10% H2, 5% CO2, and 85% N2. Or C, ambient room air enriched with 10% H2.
The strict, highly combustible anaerobic recipe requires the massive nitrogen blanket. It's B, 10% hydrogen, 5% CO2, 85% nitrogen.
Correct. Distractor A describes the microaerophilic or carbenophilic conditions required for finicky organisms like Campylobacter or Neisseria, but it still contains massive amounts of lethal oxygen.
Distractor C is a nonsensical and highly dangerous mixture. Ambient air contains 21% oxygen, which would not only be lethal to strict anaerobes regardless of added hydrogen, but in the presence of 10% hydrogen and a palladium catalyst, it represents a significant explosive hazard.
Okay, we've covered the rules, the tools, the physics, and the chemistry. Now it is time to dive into the actual bugs. We are starting with the heavyweights, the most common, most destructive organisms in the hospital. Section 8. Gram-positive Kochi, focusing on staph and strep.
These organisms are the absolute bread and butter of any clinical lab. You must be able to differentiate them immediately and flawlessly. The primary biochemical branching point for all gram-positive Kochi is the catalase test.
You drop hydrogen peroxide on a colony. If it immediately violently bubbles, converting the peroxide into water and oxygen gas, it is catalase positive.
The entire Staphylococcus genus is catalase positive. Morphologically, on a Gram stain, they classically arrange themselves in irregular three-dimensional grape-like clusters.
So we have a catalase positive, gram-positive coccus in clusters. It's a staph, Now we have to figure out which staph it is, because one of them is a killer and the others are mostly harmless skin flora.
The most important highly pathogenic species is Staphylococcus aureus. And the definitive test that separates the dangerous S. aureus from the rest of the pack is the coagulase test. S. aureus is coagulase positive.
Exactly. S. aureus produces a highly evolved enzyme that literally hijacks the human coagulation cascade. It converts soluble fibrinogen in human plasma into a solid fibrin clot.
The bacteria wraps itself in this human fibrin shield, completely hiding from our immune system's white blood cells. S. aureus is a major aggressive pathogen. It causes classic, painful, purulent skin infections. Furuncles, boils, deep carbuncles, and bullous impetigo.
It causes paronychia, which are aggressive nail bed infections, and it is a leading cause of severe post-surgical wound infections and deadly bacteremia.
But it's not just the direct invasive tissue infections. S. aureus is terrifying because it produces an arsenal of exotoxins that cause severe systemic intoxications, even if the bacteria itself hasn't invaded the bloodstream.
Yes, the toxin profile is heavily tested. It produces exfoliatin toxin, which chemically cleaves the stratum granulosum layer of the epidermis, causing scalded skin syndrome, primarily seen in vulnerable neonates.
It produces TSST1, toxic shock syndrome toxin. This is a superantigen. Normally, a T cell only activates if it finds a perfect specific match. TSST1 acts like a biological staple gun, indiscriminately locking macrophages and T cells together, forcing a massive systemic release of cytokines that violently drops blood pressure to zero.
And finally, it produces a heat-stable enterotoxin that causes rapid onset food poisoning. The text specifically notes that violent symptoms appear just 1 to 5 hours after eating contaminated foods, like potato salad or cream dishes left in the sun.
Because you aren't waiting for the bacteria to slowly grow in your gut, the bacteria already grew in the potato salad, pumped out the toxin, and you're having a massive physiological reaction to the preformed toxin you just swallowed.
And clinically, you must know that most strains of S. aureus are highly resistant to penicillin due to the production of a plasmid-mediated beta-lactamose enzyme.
Okay, I promise zero fluff, but the text includes these amazing virulent versus mnemonics to help memorize these dense profiles, and I'm going to perform the one for S. aureus right now to lock these traits into your brain.
Let's hear it.
Staph aureus is my nickname. In the Pathogen Hall of Fame, look for bubbles with catalase and for clots with coagulase. Gram-plus clusters everywhere, skin and gut, nose and hair. Protein A is my main trait. And enterotoxin in what you ate, penicillin won't touch me. Methicillin, well, maybe vancomycin killed in days of yore, but now that drug is not for sure.
It's surprisingly comprehensive. It hits the gram stain, the catalase, the coagulase, protein A, which binds the FDAC portion of human IgG antibodies upside down to avoid phagocytosis, the enterotoxin, and the escalating antibiotic resistance profile from penicillin to methicillin to vancomycin. Very effective. Now, what if the staph bubbles with catalase, but it is coagulase negative?
These are collectively referred to as coagulase negative staph, or CONANS. They are generally opportunistic pathogens. They live on our skin safely until they find an artificial entry point. The two most clinically relevant species you must differentiate are Staphylococcus epidermidis and Staphylococcus saprophyticus.
And you differentiate them using a specific antibiotic disc, novobiocin.
S. epidermidis is notorious for forming thick, impenetrable biological slimes, biofilms, on artificial plastic and metal surfaces. It causes severe endocarditis on artificial heart valves and chronic infections on prosthetic joints and IV catheters. Crucially, in the lab, it is sensitive to novobiocin. It produces a large zone of inhibition.
S. saprophyticus, on the other hand, does not typically infect plastic. It is a classic, highly specific cause of acute urinary tract infections, particularly in young sexually active females. And biochemically, it is entirely resistant to novobiocin. The bacteria grows right up to the edge of the disc.
Okay, that covers the catalase positive side of the family tree. Now let's look at the organisms that do not bubble. The catalase negative side, specifically the Streptococcus genus. Morphologically, on a gram stain, strep classically appears in spherical or oval pairs and long chains rather than clustered grapes.
The beta hemolytic streptococci, the ones that completely shatter the red blood cells on the agar, creating a clear yellow halo, are further categorized by Lancefield grouping, which is based on the highly specific antigenic characteristics of the C carbohydrate in their cell wall.
The most feared aggressive member here is Group A strep, Streptococcus pyogenes. It is strongly beta hemolytic.
It owes that violent hemolysis to two specific exotoxins, streptolysin S, which is oxygen stable, and streptolysin O, which is oxygen labile and highly antigenic, meaning the human body mounts a massive antibody response to it.
S. pyogenes also produces the erythrogenic toxin responsible for the full-body rash of scarlet fever, which is exactly what the Dick test and Schultz-Charlton test are identifying.
It is the primary cause of acute pharyngitis strep throat, as well as highly contagious skin infections like impetigo. But the real danger is that if left untreated, the antibodies your body makes to fight the strep can accidentally cross-react with your own heart valves and joints, leading to severe autoimmune sequelae like rheumatic fever.
The key identification test in the lab. It is exquisitely sensitive to a low-dose 0.04 unit bacitracin disc. And notably, unlike staph, it remains universally highly sensitive to basic penicillin.
Time for the second virulent verse. Here is the mnemonic for S. pyogenes. Streptococcus pyogenes penicillin will kill with ease, but protein M is the biggest trick to make throat and muscles sick. Antistreptolysin O will lay the heart and kidneys low. Bacitracin is my sign, zone of inhibition every time. See what I do on BAP, destroy the sheep RBC.
Again, it brilliantly hits all the high-yield points. Penicillin sensitivity, the M protein virulence factor, which repels white blood cells, the ASO antibodies causing post-streptococcal glomerulonephritis and rheumatic fever, bacitracin sensitivity for lab ID, and massive beta hemolysis on blood agar. Excellent. Now, moving to the other major beta hemolytic threat, group B strep, Streptococcus agalactiae.
S. agalactiae produces a much narrower, subtle zone of beta hemolysis. Clinically, it is a major terrifying cause of neonatal sepsis and meningitis. It frequently colonizes the vaginal tract of healthy women. It can affect the infant during childbirth, which is why pregnant women are routinely screened for it.
The laboratory identification relies on two key biochemical tests. First, it produces a positive CAM-MP reaction. This is a fascinating synergistic test. If you streak S. agalactiae perpendicular to a streak of S. aureus, there are two different toxins interact where they meet, producing a massively enhanced, distinct arrowhead of complete hemolysis. Secondly, it possesses the enzyme to hydrolyze sodium hippurate, making it hippurate positive.
Next is group D strep. This group is split into two distinct clinical categories, the Enterococcus group and the non-Enterococcus group.
Both groups share a fundamental hardiness. They will both grow in the presence of 40% bile, and they both hydrolyze the chemical esculin, making them strongly bile-esculin positive, turning the agar black. The key differentiator between the two is extreme salt tolerance. The Enterococcus species, which are notorious for urinary tract infections and high-level antibiotic resistance, will grow robustly in 6.5% sodium chloride broth. The non-Enterococcus group D species, like Streptococcus galolyticus, will not grow in 6.5% salt.
Okay, leaving the Lancefield beta hemolytic groups, we have the alpha hemolytic streptococci. These don't shatter the red blood cells, they just bruise them, turning the agar a murky, dark olive green due to the oxidation of hemoglobin to methemoglobin. The most important deadly organism here is Streptococcus pneumonia.
The colony morphology on blood agar is highly distinctive. They form alpha hemolytic, crater-like, or water drop colonies with depressed centers as they age. If the strain possesses a thick, protective polysaccharide capsule, the colonies will appear extremely wet and mucoid. On a gram stain, they don't form long chains. They are classic lancet-shaped diplococci that look like two little flames touching base to base.
Clinically, it is the leading cause of lobar pneumonia, classically presenting in board questions with a rusty-colored, blood-tinned sputum. It's also a major aggressive cause of meningitis, bacteremia, and otitis media in children.
For definitive lab ID, you perform the Quellung reaction. Quellung is German for swelling. You mix the bacteria with specific antibodies. If they bind, the capsule appears to massively swell under the microscope, confirming its presence. Biochemically, pneumococcus is uniquely sensitive to the chemical opticin, creating a large zone of inhibition. And it is highly bile-soluble. If you drop bile salts directly onto the colony, the bacteria literally dissolve and disappear. Also, you must aggressively check its sensitivity to penicillin using an oxacillin disc. A zone greater than 20 millimeters indicates it is safely sensitive.
The remaining alpha hemolytic streps are lumped into the viridins group. They are normal oral flora, but they are notorious for entering the bloodstream during dental procedures and causing subacute bacterial endocarditis on damaged heart valves. They are resistant to opticin and are not dissolved by bile.
We have an important remember elephant box right here in the text summarizing the major biochemical splits.
Let's read the elephant's mind. Staph equals clusters, catalase positive, and can survive and grow in high 7.5% salt. Strep equals pairs or chains, catalase negative, and only the hardy Enterococcus group can grow in moderate 6.5% salt.
And the specific test alerts for this section are massive and explicitly demand mastery. They want you to focus heavily on susceptibility testing of staph, specifically looking out for the beta-lactamase production that destroys penicillin and the novobiosin resistance that defines S. saprophyticus. They want you to know the specific organism that legally does not require routine sensitivity testing, which is S. pyogenes, because, astonishingly, it remains universally susceptible to penicillin. They want you to know the specific growth requirements of strep, and they strongly emphasize knowing the key defining characteristics of pneumococcus, the optogen sensitivity and the bile solubility.
All right, that was a huge, dense section. Let's run the board drill and test that complex deductive reasoning. Listener, you are up.
Scenario 1. A 22-year-old female presents to the outpatient clinic with acute dysuria, frequency, and suprapubic pain indicative of a urinary tract infection. The urine culture yields a pure growth of a gram-positive coccus growing in irregular clusters. The laboratory technologist performs rapid biochemical testing and determines the isolate is strongly catalase positive, aggressively coagulase negative, and grows right up to the edge of a novobiosin disc, indicating complete resistance. Based on this precise profile, the causative organism is definitively A. Staphylococcus epidermidis, B. Staphylococcus saprophyticus, or C. Staphylococcus aureus.
We break down the clues. Gram-positive coccus in clusters, catalase positive. We are definitively in the Staphylococcus genus. Coagulase negative means we eliminate the heavy hitter. It's a CON-S, a novobiosin-resistant UTI in a young, sexually active woman. That is the absolute classic textbook presentation for B, Staphylococcus saprophyticus.
Perfect clinical reasoning. Distractor A, S. epidermidis, is indeed a coagulase-negative staph, but it is biochemically novobiosin sensitive and is typically associated with indolent infections of prosthetic devices, central lines, and artificial valves, not acute community-acquired UTIs in young, healthy women. Distractor C, S. aureus, is instantly ruled out the moment the coagulase test resulted as negative.
Let's hear scenario 2.
A pregnant woman at 36 weeks gestation undergoes a routine rectovaginal swab screening. The swab is inoculated into a selective broth and subsequently plated on blood agar. The laboratory isolates a gram-positive coccus in short chains that produces a very narrow, subtle zone of beta-hemolysis. Further testing reveals the isolate produces a highly positive CAMP-EP reaction, demonstrating a classic arrowhead of synergistic hemolysis when streaked perpendicular to S. aureus, and it is also positive for sodium piperate hydrolysis. This organism, which poses a severe risk for neonatal meningitis, is A. Streptococcus pyogenes, B. Streptococcus agalactiae, or C. Enterococcus faecalis.
The CAMP test arrowhead and the hippurate hydrolysis are the defining gold standard characteristics for identifying group B strep. And group B strep is the major neonatal threat. The answer is B, Streptococcus agalactiae.
Exactly. Let's review why the others fail the biochemical panel. Distractor A, S. pyogenes, or group A strep, is characterized by a massive zone of beta hemolysis and is identified by its extreme sensitivity to a low-dose basotracin disc, not by the CAMP or hippurate tests. Distractor C, Enterococcus faecalis, is uniquely identified by its ability to hydrolyze bile esculin, turning the agar black, and its robust ability to grow in 6.5% sodium chloride broth.
Final scenario for the gram-positives.
You are examining a blood agar plate inoculated with purulent, blood-tinged sputum from a 70-year-old patient with severe acute respiratory distress. You observe distinct alpha-hemolytic crater-like colonies with depressed centers. A gram stain reveals gram-positive, lancet-shaped diplococci. Confirmatory testing reveals the organism is exquisitely optogen-sensitive and rapidly dissolves when exposed to bile salts. What specific clinical disease state is most classically associated with this organism? A, acute lobar pneumonia with rusty sputum, B, post-infectious rheumatic fever, or C, neonatal sepsis and meningitis.
We have to identify the bug first. Alpha-hemolytic, lancet-shaped diplococci, optogen-sensitive, and biosoluble. That precise combination belongs exclusively to Streptococcus pneumonia. The classic board-tested disease presentation it causes is A, acute lobar pneumonia with rusty sputum.
Spot on. You must link the biochemical ID to the clinical presentation. Distractor B, rheumatic fever, is a severe delayed autoimmune post-infectious sequela of Streptococcus pyogenes, group A strep, not pneumococcus. Distractor C, neonatal meningitis, is classically associated with vertical transmission of group B strep, S. agalactiae, or the cold-loving Listeria monocytogenes, not community-acquired pneumococcus.
Moving on, we are leaving the robust gram-positive purple grapes and chains behind. We are flipping the gram stain pink, and we are examining the delicate kidney bean-shaped organisms. Welcome to Section 9, gram-negative Kachi, focusing on the pathogenic Neisseria and Moraxilla species.
The key identifying characteristics that unite this entire group are critical to recognize instantly. Morphologically, on a gram stain, they appear as pink gram-negative Diplocaci, but they aren't perfect spheres. They are classically described as kidney bean-shaped or coffee bean-shaped with their flat sides facing each other. Biochemically, the entire group produces the cytochrome oxidase enzyme, making them strongly oxidase positive. They will rapidly turn a specialized swab deep purple.
The two major terrifying pathogens here are Neisseria gonorrhea and Neisseria meningitis. Let's start with the GCN gonorrhea.
It is an incredibly fastidious, delicate organism. It will not grow on MacConkey agar, and it won't even grow on standard blood agar. It strictly requires the pre-lelized red blood cells of chocolate agar, or the highly selective Thayer-Martin agar we discussed earlier to suppress normal flora. It requires an enriched 5 to 10% CO2 capnophilic environment, and it is painfully slow growing, often taking a full 48 hours for tiny colonies to appear.
The biochemical hallmark and the absolute most testable fact used to differentiate the species is its sugar fermentation profile, utilizing cysteine trypticase agar sugars. N. gonorrhea ferments GL-Eucose only. It ignores maltose, lactose, and sucrose.
Clinically, it causes the sexually transmitted disease gonorrhea, which can present as severe purulent urethritis, cervicitis, or systemic disseminated joint infections. It may be completely asymptomatic, especially in females, acting as a silent reservoir, and is frequently seen as a clinical co-infection with chlamydia trachomatis. An incredibly important laboratory and legal note. Observing a direct patient gram stain showing intracellular gram-negative Diplocaci actively being phagocytosed by neutrophils is considered highly sensitive and diagnostically definitive for symptomatic males. However, it is not considered sensitive or definitively diagnostic for females because the normal vaginal tract contains harmless Neisseria and Acinetobacter flora that can perfectly mimic GC under the microscope. You must wait for the culture.
And we must reiterate the golden pre-analytical rule. Do not refrigerate the suspected GC specimen prior to culture or the cold shock will kill it. Also, because plasmid-mediated resistance is now rampant, you must routinely perform a rapid beta-lactamase test on the isolate to determine if penicillin therapy will fail.
Time for the Neisseria virulent verse. Let's lock in the profile. Gonorrhea the clap, GC. I'm an old-fashioned STD. I need CO2 and chocolate agar and may not grow for 48 hours. I ferment glucose, the only sugar for me. Do an oxidase in purple, you'll see.
An excellent, concise summary of the critical diagnostic parameters. Now, we must contrast that with its legal cousin, Neisseria meningitidis.
N. meningitidis is slightly less fastidious. While it prefers chocolate and Thayer-Martin, it will often grow on standard blood agar as well. The crucial defining biochemical difference is the sugar fermentation pattern. While gonorrhea ferments only glucose, meningititis ferments both G-locose and maltose.
It is highly contagious, transmitted by respiratory droplets in close quarters like college dormitories or military barracks. It causes devastating rapid-onset bacterial meningitis. A severe, life-threatening complication the boards expect you to recognize is Waterhouse-Friderichsen syndrome. This occurs when a massive systemic meningococcal infection triggers disseminated intravascular coagulation. It is physically characterized by a rapidly spreading, scattered petechial rash across the body and catastrophic, massive hemorrhage into the adrenal glands, leading to acute adrenal failure and death.
The third organism commonly grouped in this section is Moraxilla catarrhalis, which was formerly classified under the genus Bronamella. It is a common cause of respiratory infections, sinusitis, and otitis media in children. It grows very well in standard chocolate and blood agar, but importantly, it does not grow on MacConkey's agar despite being a gram-negative organism.
The text highlights a very specific, physical, almost funny behavioral quirk of Moraxilla colonies on an agar plate.
Yes, the classic hockey puck sign. The colony structure is incredibly stiff and cohesive. If you take an inoculating loop and try to push the colony, it doesn't smear like normal bacteria. It remains completely intact and literally moves over and slides across the agar surface like a puck on ice.
Biochemically, how do we definitively distinguish this hockey puck from the dangerous Neisseria species?
Moraxilla catarrhalis is completely asaccharolytic, meaning it lacks the enzymes to ferment any carbohydrates at all. It is negative for glucose, negative for maltose, and negative for lactose. Also, be clinically aware that the vast majority of strains are strongly beta-lactamase positive, meaning ampicillin and penicillin are useless against them.
And we have another remember box right here in the text, summarizing the exact sugar differentiation, which is the absolute key to surviving this section on the exam. Let's review the progression. N. gonorrhea is positive for glucose only. N. meningitis is positive for glucose and maltose. And they throw in a third species here, N. lactamica, which is harmless, normal flora found in the respiratory tract, but can be easily confused with pathogens on a plate. It is positive for glucose, maltose, and lactose.
Glucose, maltose, lactose. It's a perfectly easy additive progression to memorize. Let's hit the board drill for section 9. Listener, back to the hot seat.
Scenario 1. An oxidase-positive, kidney bean-shaped gram-negative Diplococcus is rapidly isolated on chocolate agar from the cloudy CSF of a young adult patient presenting with a stiff neck, fever, and a petechial rash. Rapid biochemical CTA sugar testing shows that the organism actively ferments both glucose and maltose, but strictly does not ferment lactose. Based on this precise biochemical profile, the organism is definitively A. Neisseria gonorrhea, B. Neisseria meningitis, or C. Neisseria lactamica.
Positive for glucose and maltose. That two-sugar profile points directly and exclusively to B. Neisseria meningitis.
Correct. The sugar fermentation profile is the definitive identifying test here. Distractor A, N. gonorrhea, is incorrect because it is metabolically restricted and ferments only glucose. Distractor C, N. lactamica, is incorrect because it possesses the enzymes to ferment lactose in addition to glucose and maltose.
Let's hear scenario 2.
A technologist is working up a sputum culture from a patient with a severe exacerbation of COPD. They observe a predominance of gram-negative Diplococci. The colonies growing on the blood agar are highly distinctive. They are described as remarkably hard, and they move over entirely intact when pushed with an inoculating loop. Further biochemical testing reveals the organism is completely asaccharolytic. The organism is A. Moraxilla catarrhalis, B. Neisseria gonorrhea, or C. Acinetobacter baumannii.
The physical hockey puck sign combined with the absolute inability to ferment any sugars. That is the classic, heavily tested description of A. Moraxilla catarrhalis.
Yes. Distractor B, N. gonorrhea, ferments glucose and produces soft, normal colonies that do not exhibit the hockey puck morphology. Distractor C, Acinetobacter baumannii, is mentioned in advanced texts as a plump gram-negative coccobacillus that can be morphologically confused with Neisseria on a hurried gram stain. But it does not exhibit the moves-over trait or the specific robust oxidase-positive diplococcal profile we are discussing.
Final scenario for the gram-negative Cocci.
A busy outpatient clinic sends a cervical swab to the laboratory for culture, highly suspecting an active Neisseria gonorrhea infection. The laboratory strictly utilizes the highly selective Thayer-Martin agar to isolate this pathogenic Neisseria from the heavy mix of non-sterile vaginal flora. To ensure the viability of this incredibly delicate pathogen during transport, what must the clinic staff never do to the suspected GC specimen prior to culturing it? A. Expedite it to the laboratory to be set up within 2 hours. B. Place it in the specimen refrigerator to preserve it overnight. Or C. Utilize a transport system that generates a 5–10% CO2 environment.
This goes directly back to our fundamental pre-analytical rules. You must never refrigerate a GC specimen. The thermal cold shock physically shatters their membranes and kills them. Answer B.
Correct. Refrigeration will almost certainly result in a false negative culture, masking the STD. Distractor A is a correct and necessary action. You absolutely should set it up as quickly as possible to prevent normal flora overgrowth. Distractor C is also a highly correct and necessary action. They require a CO2-enriched capnophilic environment for optimal survival and growth.
Excellent. We've conquered the Cocci. Now let's tackle the robust, diverse, and occasionally terrifying class of the gram-positive rods. This is section 10, and it is packed with unique morphologies.
This is a highly disparate group of organisms. Let's start with the agent of diphtheria, Corynebacterium diphtheria. Morphologically, under the microscope, it is very distinct. On a gram stain, you see small, highly pleomorphic, meaning they vary wildly in shaped rods with heavily clubbed, swollen ends. Due to their unique snapping method of cellular division, they range themselves at sharp angles, classically described by the boards as a palisade or a Chinese letter formation.
And if you stain them specifically with methylene blue dye after growing them on Loeffler's serum medium, you can visualize large, dense, metachromatic granules inside the cells. These are essentially massive storage depots of polyphosphate, and they strangely stain a deep red-purple against the blue cell body.
For selective isolation from a throat swab, you utilize Tinsdale agar or cysteine tellurite blood agar. C. diphtheria produces characteristic dark grayish-black colonies on this media due to its ability to enzymatically reduce the tellurite salt.
Clinically, it causes respiratory diphtheria, which is characterized by a thick, leathery, suffocating gray pseudomembrane forming on the tonsils, uvula, or soft palate. It can physically block the airway, but the bacterium sitting in the throat isn't what's truly dangerous, right? It's the systemic toxin it pumps out into the bloodstream.
Exactly. The lethal systemic disease, which attacks the heart muscle and peripheral nerves, is exclusively mediated by the diphtheria exotoxin. And here's a fascinating, highly testable microbiological fact. The bacteria itself doesn't possess the genetic code for the toxin. It only produces this deadly exotoxin if it has been previously infected by a specific carrier bacteriophage, known as a B phage, that inserts the toxin gene into the bacterial chromosome. This is called a lysogenic conversion. If the bacteria isn't infected by the virus, it is harmless. Therefore, isolating the bacteria isn't enough. To prove the isolate is actually dangerous, you must use the ELEC test, which determines in vitro via immunodiffusion if that specific bacterial colony is actively producing the lethal toxin.
Incredible biology. A virus weaponizes a bacteria against a human. Moving on to another incredibly unique, dangerous pathogen, Listeria monocytogenes.
Listeria produces small, grayish colonies on blood agar with a very narrow, subtle zone of beta hemolysis. Because of this appearance and its tendency to sometimes look like short Cocci on a gram stain, it can easily be misidentified as group B strep. However, biochemically, it is catalase positive, and it is strongly bile-esculin positive, turning the slant black.
But the defining physical feature, the absolute golden ticket the boards love to test, is its bizarre temperature-dependent motility profile.
In a wet mount preparation examined under the microscope, it exhibits a rapid end-over-end tumbling motility. But more specifically and testably, if you inoculate it deep into a tube of semi-solid sim-motility media, it produces a highly characteristic umbrella pattern of growth slightly below the surface. But here is the massive biochemical trick. It only expresses its flagella and produces this umbrella motility when incubated at room temperature, around 25 degrees Celsius. It actively downregulates its flagella and does not produce this motility when incubated at human body temperature, 37 degrees Celsius, presumably to evade detection by the human immune system.
And as we discussed in the special media section, it thrives in cold temperatures, which is why we use 4-degree cold enrichment to selectively isolate it from heavily contaminated food or stool samples. Clinically, it's a major cause of devastating neonatal meningitis, sepsis, and stillbirths, most often acquired by pregnant women eating contaminated, unpasteurized dairy, soft cheeses, or cold-processed deli meats.
Let's do the virulent verse for Listeria to lock all of that in. I'm Listeria, cold temps my game. Umbrella motility brings me fame. I'm a gram-positive rod and hemolytic. Kills baby in utero after mom gets sick from eating dairy or processed meat or veggies not exposed to heat.
A perfect, concise encapsulation of every testable fact. Next on the list of gram-positive rods is the unique Erysipelotheryx rhusiopathiae.
This one is an outlier. It is a thin, non-modal, catalase-negative rod.
The morphological key for identifying Erysipelotheryx is how it grows physically in a tube of gelatin media. It radiates outward from the stab line, producing a highly specific pattern that looks exactly like a bristly test tube brush.
And biochemically, it has one massive, glaring, distinguishing feature among all the gram-positive rods.
Yes, it is the only clinically significant gram-positive rod that is strongly H2S positive, producing massive blackening in the butt of a TSI agar slant due to the production of hydrogen sulfide gas. Clinically, you absolutely need to know the occupational association. It causes a painful, localized skin infection called erysipeloid. It is almost exclusively seen in commercial fishermen, butchers, veterinarians, and rose growers who sustain deep puncture wounds on their hands while handling raw animal products, fish scales, or thorny plants contaminated with infected animal feces.
Finally, we end this section with the big, tough spore formers. The genus Bacillus.
As a genus, Bacillus species are massive, thick, gram-positive to gram-variable rods that form highly resilient, distinct internal endospores. Because they are so robust, they generally produce large, spreading, flat colonies on agar that look like frosted grau glass, and they are vigorously catalase positive.
The most notorious, historically feared member is Bacillus anthracis.
On a gram stain, the thick rods form very long, perfectly aligned chains, classically described by the boards as looking like rigid bamboo shoots. On the agar plate, the edges of the colonies swirl outward in long, curling filaments, giving them a highly characteristic Medusa head appearance. Crucially, unlike almost all other common environmental Bacillus species, the deadly B. anthracis is completely non-modal, and it is completely non-hemolytic on sheep blood agar.
Clinically, it is a Category A bioterrorism agent. It causes anthrax, which can present in three forms: cutaneous anthrax forming a black Escher wound, highly fatal pulmonary anthrax from inhaling spores, or severe gastrointestinal anthrax from eating infected meat. And we must contrast that severe lethal pathogen with its much milder common cousin, Bacillus cereus.
Bacillus cereus is typically strongly beta-hemolytic and highly motile, which easily differentiates it from anthracis. Clinically, it is famous for causing a rapid onset, violent food poisoning due to the ingestion of a preformed enterotoxin. The classic board association is fried rice syndrome. The tough spores survive the initial rapid boiling of the rice. If the rice is left sitting out at room temperature at a buffet, the spores germinate into vegetative bacteria, multiply rapidly, and pump out massive amounts of heat-stable enterotoxin. When the rice is briefly reheated and eaten, the bacteria die, but the toxin survives and causes violent vomiting within hours.
Incredible. Biology is a terrifying, fascinating arms race. Okay, listener, let's run the final standard board drill for Section 10. Put all those clues together.
Scenario 1. A 45-year-old commercial fisherman presents to the clinic with a painful, swollen, deeply erythematous lesion on his hand, sustained after a puncture wound from a fish spine. A swab of the lesion yields a thin, gram-positive rod. In the laboratory, the isolate exhibits a distinct test tube brush growth pattern when inoculated into gelatin media, and it is strongly H2S positive, turning the butt of a TSI slant pitch black. The organism is A. Corynebacterium diphtheria, B. Listeria monocytogenes, or C. Erysipelothryx rhizopatheae.
The clinical history of occupational exposure to fishermen, combined with the test tube brush morphology and the absolute clincher, the only H2S positive gram-positive rod. That is definitively C. Erysipelothryx.
Correct. The distractors are easily and rapidly eliminated by the biochemical profile. Distractor A, Corynebacterium, produces dark black colonies on Tinsdale agar due to tellurite reduction, but it is strictly H2S negative in TSI media. Distractor B, Listeria, exhibits umbrella motility in SIM media and is also H2S negative.
Scenario 2.
A patient presents to the hospital emergency department with severe acute symptoms of violent nausea and vomiting. The symptoms began just 2 hours after eating leftover fried rice from a local buffet. The public health laboratory investigates the food source and isolates a massive gram-positive rod that clearly contains internal endospores. The colonies are wildly beta-hemolytic on blood agar and are highly motile. Which specific organism is most likely responsible for this intoxication? A. Bacillus cereus, B. Bacillus anthracis, or C. Listeria monocytogenes.
Leftover fried rice, resilient spores, rapid onset from a preformed toxin, and is beta-hemolytic. That classic profile is A. Bacillus cereus.
Yes. Distractor B, Bacillus anthracis, is a spore-forming rod, but it is incorrect because it is classically completely non-hemolytic, non-motile, and causes severe systemic anthrax, not rapid onset emetic food poisoning associated with rice. Distractor C, Listeria, is incorrect because it is a very small rod that physically does not form endospores, and its classic foodborne association is with unpasteurized dairy and cold deli meats, not rice.
Last scenario before the lightning round.
You observe small, gram-positive pleomorphic rods arranged in a sharp Chinese letter palisade formation from a throat swab of a patient with a thick pharyngeal pseudomembrane. You suspect the organism is Corynebacterium diphtheriae. To legally and definitively confirm that this specific patient isolate is actively producing the dangerous exotoxin in vitro, you would order which of the following specialized tests? A. The Schick test, B. The ELEC test, or C. A Loeffler's methylene blue stain.
The organism is presumed to be C. diphtheriae. But to prove the specific isolate carries the lethal phage and produces the toxin in vitro in the lab, we use the immunodiffusion assay, B. The ELEC test.
Correct. The ELEC test is the definitive in vitro precipitation assay for active toxin production. Distractor A, the Schick test, is incorrect because it is an in vivo test injected directly into a human patient's skin to determine their systemic immunological susceptibility to the disease, not to test a laboratory isolate. Distractor C, Loeffler stain, is used merely to enhance the microscopic visibility of the intracellular metachromatic granules to help identify the organism morphologically. But a stain absolutely cannot prove physiological toxin production.
That is 10 dense, heavily fact-filled sections. You have survived the barrage. But before we let you go, we are going to do the lightning round. A rapid-fire final pass pulling only the absolute highest yield, most likely to be tested facts from the entire document, every single item specifically flagged with a test alert box. We're going to sprint through these and solidify exactly why the board examiners love to test them.
Let's do it. First up, autoclave specifications. 15 psi, 121 degrees Celsius for 15 minutes. The boards love this because sterilization is a life or death absolute in the hospital. If you don't know the exact physical parameters to achieve the destruction of endospores, you are dangerous in a laboratory setting.
Next, Kirby-Bauer disc diffusion preparation rules. It must be Mueller-Hinton agar, poured to exactly 4 millimeter depth, with a pH strictly between 7.2 and 7.4. And the bacterial inoculum must precisely match a McFarland 0.5 standard, which is 1.5 times 10 to the 8th organisms. The boards test this relentlessly because disc diffusion is entirely dependent on the standardized physics of molecular movement. If you change the agar depth or the bacterial concentration, the zone sizes change, the results become clinically meaningless, and you might tell a physician to give a patient a drug that will fail.
The confusing tests. Know the absolute difference between the ELEC test and the Schick test. ELEC is an in vitro plate test to prove the isolate's toxin production. Schick is an in vivo skin injection to prove the patient's susceptibility. They test this because the names are obscure, old, and easily jumbled, which perfectly separates the students who just blindly memorize terms from the technologists who truly understand the immunological applications.
Blood culture dilution. You must maintain a strict 1:10 ratio of patient blood to liquid broth. Why? Because human blood is a weapon full of complement proteins, circulating antibodies, and white blood cells that are actively trying to kill the bacteria you are trying to grow. If you don't dilute those factors out with a massive volume of broth, you'll get a false negative culture and you will miss a fatal bloodstream infection.
Susceptibility testing of Staphylococcus. You must focus intensely on detecting beta-lactamase production to immediately rule out penicillin therapy, and you must know the novobiosin resistance profile to quickly distinguish the UTI-causing S. epiphyticus from the prosthetic infecting S. epidermidis. They test this because staph is absolutely everywhere in the hospital, and missing a resistant strain leads to immediate, catastrophic treatment failure.
The organism not requiring sensitivity testing, that is, Streptococcus pyogenes, the group A strep. Why is it a test alert? Because decades after the discovery of antibiotics, and despite massive global resistance trends, it remains miraculously and universally susceptible to basic penicillin. If you waste laboratory time and hospital money running a full, expensive MIC panel on a routine strep swab, you aren't practicing efficient, knowledgeable clinical microbiology.
And finally, the key defining features of pneumococcus, or S. pneumonia. It is uniquely optogen-sensitive and rapidly bile-soluble. The boards love this because it perfectly, easily, and cheaply distinguishes a highly lethal meningitis-causing pathogen from the relatively benign normal flora of the viridins strep group that share its exact same alpha-hemolytic green appearance on a blood agar plate.
And there it is. We have drilled the foundational growth requirements, the physics of sterilization techniques, the molecular mechanisms of antibiotics, and the intricate biochemical profiles of the major gram-positive and gram-negative coaches and rods. You have navigated the diagnostic muddy waters. You understand the why and the how, not just the what. You are ready.
We spend so much time in the lab memorizing how to kill these bacteria with autoclaves, heavy metals, and complex antibiotics, but look at the sheer genetic elegance of what we are fighting. Staphylococcus aureus effortlessly acquiring a plasmid to build an enzyme that destroys our best beta-lactam drugs. Or Corynebacterium diphtheria, a basic rod, only producing its deadly systemic toxin when it is itself infected and reprogrammed by a viral phage. You're not just studying static diseases, you're studying the most adaptable, resilient, rapidly evolving survivors on the planet. Next time you see a medusa head colony swirling on a plate, ask yourself, what complex, millennia-long evolutionary arms race produced that?
That is fantastic, humbling thought to leave on. Thank you for diving deep with us today. Keep reviewing the logic and the mechanisms behind the tests, not just the answers. We'll catch you on the next deep dive.